Hello, good morning. Hello. Check. Hello. Good morning. Am I visible? Audible? Good morning all. So how are you doing all? So in last class, hope you have revised your last class. Okay. Myself Dr. Amit Jain. So I'll be taking your rest of the lectures in biochemistry. Okay. So last it was amino acid that you have started, right? Okay, amino acid was just started and only classification was done, right? Only the classification was done. So now we'll start with the next topic, and that is the catabolism of amino acids, or simply the metabolism of amino acids. Okay. So we'll continue with that, then we'll take heme metabolism, then we'll take the next nucleotide metabolism, and then we'll take the genetics part. Okay, so you have 4 sessions including this one, right? 7, 8, 9 and this session also. Okay, so hope you have started your revision part. Okay, so sooner you revise, better you retain, as you all know, isn't it? Okay. So there are a lot of things that you have to see apart from SSR case, right? So just don't stuck to that. Okay. So I'm starting with amino acids, right? So let's start. Amino acids, right? So what are amino acids that you have already done? So something which has amino group and acidic group, that's called as amino acid. Okay, and then you have read about the classification. Okay, so then we have some aromatic amino acids like phenylalanine, tryptophan, tyrosine. Okay, then we have amino acids which are branch chain like valine, isoleucine and leucine. Okay, then we have sulfur containing amino acid methionine, and as such groups, as such group containing amino acids are cysteine, right? As such group, that is sulfhydryl group. Okay, so that one also. Okay, then we have amino acids which are highly hydrophobic, like most hydrophobic amino acid is isoleucine, where it is present. So hydrophobic amino acids are mostly present in the like interiors, right, away from the water. Like this is the cell membrane, so hydrophobic amino acid will be present in the interior. That you have all done, so I'm just revising the basics of amino acid. Okay, so isoleucine, leucine and valine, these are the 3 most highly hydrophobic and also the branch chain amino acids. Fine. Then we have amino acids in chemistry part again, that's neutral, basic, acidic. So guys, those which are acidic and basic, as you know, acidic are aspartic acid, glutamic acid, right? Basic are arginine, most basic. Then we have the lysine and partly histidine, not very basic. Okay, so these are the basic. So only 5 amino acids are acidic and basic, right? So rest all the 15 amino acids are actually neutral, right? So no confusion. Okay, so if someone asks you neutral amino acid, then you know what is a neutral amino acid. Okay, so these are. Then again you have the amino acids like ketogenic and glucogenic. So guys, all amino acids are glucogenic except the leucine and partly lysine. Okay, So it's a leucine, which is most, I'm going to say, the purely ketogenic amino acid. And apart from the leucine, right, you can also mark the answer as lysine. Fine. But lysine is now considered as predominantly ketogenic. So there is a new word for this, that is predominant. Okay, predominantly ketogenic. Right, so what is predominantly ketogenic? That means it is again a kind of mixed amino acid, right, as per the new reports, okay, new editions of the books. Fine. So that is your predominantly ketogenic amino acid. So that's clear. So ketogenic and glucogenic. Glucogenic something which makes glucose. So gluconeogenesis you have covered. Ketogenic means ketone body synthesis you have covered. Something which makes acetyl-CoA or acetoacetate, that's ketogenic. Right? You always make that. Okay, we'll see the metabolism later. And then we have amino acids which are essential, non-essential, so that we all know. Right. So guys, that's your that what you have learned in amino acids. Okay. Questions are very common on these topics. Right. Like which amino acid absorbs light at 280 nanometers. So what is the answer? Any answer? So the answer is tryptophan. Okay, so that is tryptophan. So like that, which amino acid has sulfidyl group? I told you cysteine. Which is the most, which are the hydrophobic that I told you, like that. Okay. So guys, catabolism. Okay. So amino acids, they are consumed in our diet every day, right? So the total protein consumption ideally should be around 1 gram per kilogram. Okay, so 0.8 to 1 gram per kilogram. Right, so that's the total consumption. Okay, just a minute. So 0.8 to 1 gram per kilogram. Okay, so like a 60 kg man required around 60 grams of the protein, right? Okay, so like that. Right. So this is the normal amount of protein that we should consume, but most of our diets, especially vegetarians, they are deficient in protein content. Okay. Fine. Now the second thing is, if you consume higher amount of protein, right, will it give you the positive nitrogen balance? No. Right. Suppose if somebody is consuming a 60 kg man is consuming 100 grams of protein, so that will not help. Right. That will not help in anything like building muscles or building or gaining higher weight. No. The only thing that will happen is excessive production of urea. Right? You know why? The problem is, you can consume high amount of carbs, right? That we do almost every day, especially in the lockdown. Right? That gives us the dopamine boost, isn't it? Okay. So high amount of carbs and fats we can consume and we can store them. Right? So we can store fats directly in the adipose tissue. We can store carbs in the form of glycogen, but that's very small quantity in the liver and muscle. So we can store rest of the carbs as fats again, right? As you know, right? We can convert carbs to the fats easily in our liver. Okay. So guys, the two things we can easily, store, but we cannot store amino acids or the proteins. Okay. Human body cannot store amino acids or proteins. So the point is, so just write the heading now. Is amino acid catabolism we are discussing. So we will discuss some disorders with special products and some important enzymes, right? So the point is that human beings cannot store nitrogen, right? So we cannot assimilate the nitrogen. Okay, we cannot store the nitrogen. So that's why we have to excrete it every day in the form of the most common form, that is urea, by the human beings, right? Urea. So that is okay. So the excess nitrogen. So when I say nitrogen, you can say we cannot store amino acids or we cannot store proteins directly. Okay, so the excess nitrogen is excreted as urea. This is the most common form. Okay, so what are the examples of nitrogen containing or the nitrogenous waste products that we produce every day? Okay, what are the examples? So guys, the most common being the urea. Second is uric acid, okay, which is also in good quantities, right? Okay, so like around 30 grams of urea we excrete in urine every day, and around 800 mg of uric acid we excrete every day in 24 hours of urine sample, right? So nitrogenous waste product. Okay, so these are the two important ones in our body, right? Fine. So human beings cannot store nitrogens. Here, remember this concept always. Okay. So, guys, one more thing: nitrogen is toxic to our body, and that is in the form of ammonia, right? Ammonia is a very toxic compound, right, which can cause the various neurological consequences that I will cover later on, right? So the excess nitrogen, so that I have written already, excess nitrogen from the amino acids that we have consumed. Okay, so somebody has consumed the excess amount of amino acids or the non-essential amino acids which may not be required, right? Okay, for example, somebody has consumed a protein which is not a very good protein and it is not rich in essential amino acids. Okay, it contains all the non-essential amino acids like alanine and glycine and etc. So those amino acids, right? Amino acids. So excess nitrogen from amino acid is transferred to the alpha ketoglutarate. So you know what is alpha ketoglutarate? You are done with your cap cycle. So there was one of the intermediate, alpha ketoglutarate. Okay. Fine. So guys, this process is known as, right? So excess nitrogen from amino acids which are not required is transferred to the alpha ketoglutarate, okay, which generate glutamate. I'll tell you, okay, glutamate and a alpha keto acid, right? And a alpha keto acid, right? So what is alpha keto acid? Some amino acid from which the amino group has been removed. That's called as alpha keto acid. Okay, it's clear? So now let's see. So guys, you know at least one or two enzymes, that is ALT and AST, right? So this process is known as, first of all, so this is known as a process called as transamination. Right? What do you call that? Transamination. Fine. So transamination, and this process transamination. So transfer of amino group, that's called as transamination. Okay, and this process need vitamin B6 dependent. So what is B6? Pyridoxine, right? So need the B6 dependent transaminase enzyme. So everything will be clear. First write. Okay. So like for example, a person has consumed alanine, which is a non-essential amino acid. So this alanine combines with alpha ketoglutarate. Okay, alpha ketoglutarate. So from the alanine which had nitrogen, now we will generate something that's called as pyruvate. So you must be knowing this already, right? So the most important amino acid for gluconeogenesis is alanine, because it generates pyruvate, which can easily enter into the gluconeogenesis process. Okay, the question was like this in the exam: which of the following amino acid is raised in the blood of a patient who is fasting for let's say 5 days? So answer is alanine. Plus, okay, it is released from the breakdown of muscle tissue and reaches liver to make glucose. Okay, so transamination process, this can be done in any tissue, right? So this can be done in any tissue, and this will generate pyruvate. So pyruvate is nothing, it is alanine minus nitrogen, and plus it generates the glutamate. So glutamic acid. Okay. So what is glutamate? Alpha ketoglutarate plus the nitrogen. Okay. Or the NH2. Instead of nitrogen, because it's amino group transferred, I can write NH2. Is it clear to all? Right. So that's your basic process, and for this we need an enzyme that's called as ALT, alanine transaminase. Okay, alanine transaminase. Fine. So guys, this can be any amino acid, right? But alpha Kg is always there. This can be any keto acid, right? Any keto acid, but this will always be glutamate, right? So these two are constant, we can say like that. Okay, so alpha Kg is always a nitrogen acceptor, right, in the process known as transamination. So very important point. Okay, alpha ketoglutarate will always be there. Similarly, like just to make you understand, suppose if I take aspartate, fine, then again I will take alpha ketoglutarate, I will take the enzyme AST, then we will generate oxaloacetate from the aspartate, the removal of amino group, plus we will get glutamate again. So these two are again constant. Clear to all? Fine. Some more points that you should know. So one thing I told you, it's all B6 dependent. So make it a rule, make it a general concept in your mind that most of the amino acid metabolism reactions are pyridoxine dependent. Mostly. I'll tell you which are not. There are few exceptions, but you can mark B6 as the answer for most of the amino acid related reaction. Okay. So that's your so we are discussing transamination, right? So this transamination, or so there's one more important point, is that this transamination reaction follows the ping pong mechanism. Okay, ping pong mechanism, right? So what is ping pong mechanism? Like, you know what is ping pong? So the ping pong mechanism is, the enzyme do not bind to the all substrates, right, before releasing the first product. Okay, what is the meaning of this? For example, this is the enzyme ALT, right? So ALT enzyme will first bind the alanine and release the pyruvate. Okay, so guys, it is not binding alpha ketoglutarate before the pyruvate has been released. Okay, are you getting the point? Okay, so that's your ping pong mechanism. What is that? That the enzyme do not, so this was once asked as a question, this name only. So enzyme don't bind all the substrates, right, before releasing, the first product. Okay, before releasing the first product. Okay, so the analogy may not be accurate, but this name has been given, right? Okay, so enzyme, first will release the pyruvate, for example, and then bind the alpha ketoglutarate. Okay, and then use a nitrogen which has been removed already from the alanine to add to the alpha ketoglutarate. Is it clear? So this name you should know. Sorry. So that's all about the transamination. So guys, what we are doing is, we are in a process of making urea. Fine. So from whatever the unnecessary amino acids that has been consumed, we will convert that into urea. First, what we are doing is a transamination reaction. Okay, transamination reaction. Okay guys, one more point is that all the excess nitrogen, right, appear as glutamate. So what is the simple meaning? That we have converted all the excess amino acids to glutamate. Is it clear, guys? Okay, fine. So all the excess nitrogen appear as the glutamate. Am I clear? So whatever we have consumed, like alanine, serine, glycine, which are non-essential amino acids, so ultimately they will all convert into the glutamate. Why? Because only glutamate can release ammonia in the liver by some various reactions, right? So all the excess nitrogen appear as glutamate, as the glutamate can undergo. So this is our next topic, that is oxidative deamination. Right, oxidative deamination. Fine, that's the one point. And lysine, proline, threonine. Okay, lysine, proline, threonine has no transaminase. So guys, there is nothing like a lysine transaminase. We have transaminase for every amino acid except these, especially lysine, proline, threonine. Okay. Okay guys, so these are some important points that I have mentioned here. So what is the second step in the urea synthesis? So this is our first topic that we have covered. Okay, it's the first topic that we have covered, and that is called as transamination. Fine. So if I have to write like this, then is urea synthesis involve 4 steps. It's not urea cycle we are discussing, urea synthesis. So first is that we are discussing, that is transamination. Okay, transamination. Second, now I will discuss, so that you remember in the sequence, oxidative deamination. Okay, 1+2 is also known as trans deamination. This is another term for 1+2. Okay, third is, so oxidative deamination will generate ammonia. You can write this will generate ammonia. Fine. What oxidative deamination? Third is ammonia transport. Why? Because ammonia is very toxic, right? It has to be removed. Okay. So, and the last 4 is the reactions of urea cycle. Okay. So as we have already covered this point, that is transamination. Fine. Then now next we'll take is oxidative deamination. So write the heading oxidative deamination. Fine, oxidative deamination. Okay. So is this process only occurs in liver? Site is liver. So guys, while reading metabolism, you must have already covered some points and you're already versed with your metabolic section, right? So while reading metabolism, you should know like what is the site, okay, where it is occurring, what is the purpose, like what is the products we are getting, and what is the actual benefit we are deriving from this. Okay, what is the product? Then you should know what is the rate limiting enzyme of this particular pathway, and then you should know that what are the vitamins used. Okay, because vitamins are coenzymes, they're activities of enzymes. Am I clear? Okay. And is there any ATP used or not? What is the regulation, etc. So these are the points you should keep in your mind while reading the metabolic pathways. Because most of the time you will be asked question, based on that point, right? Definitely the changing system of examinations are under process, right? So even if you get a clinical question, right, which is based on, let's say, a case of urea cycle disorder, the point they will ask you ultimately will be is not the diagnosis, right? But when they test you for the subjects like first year and second year subjects especially, okay, they will not test you for the diagnosis directly, they will test you for the basic knowledge, right? Definitely the question will be a clinical form, right? That I'll tell you some questions also, right, like that. But the basics will always help you. Okay, like this is the pathway, it occurs in liver, what is the purpose? Okay, it releases ammonia, and is there any ATP used? No. Okay, like that. Is there any disorder associated, any vitamin used, any regulation important for our cells, health or body, whatever, like that. Okay. So oxidative deamination site is liver. Find this. Then purpose I told you, to release the ammonia. Okay, so it releases the ammonia. Ideally ammonia that should be released in liver only, but it's not the case. Ammonia can be released in the various tissues, but most commonly oxidative deamination occurs in the liver. Fine. So how it is done? So we take the glutamate. So why glutamate? I told you, nitrogen from every amino acid. This is acted upon by glutamate dehydrogenase enzyme, that is GDH in short, right? Okay, glutamate dehydrogenase. So this releases the ammonia, okay, and obviously produces the alpha ketoglutarate. Right here, right? So glutamate dehydrogenase is the name of enzyme which generates ammonia. So what is the unique points about this glutamate dehydrogenase? Okay, so this enzyme is zinc dependent. First, this is a dehydrogenase, and guys, as you know, all the dehydrogenase needs which vitamin? Any answer? You are done with lots of dehydrogenases till now. Dehydrogenase needs which enzyme? Most commonly niacin, okay, and in the form of NAD. So guys, this glutamate dehydrogenase can use NAD to NADH because it's dehydrogenation, or it can use NADP also and make NADPH, just like the enzymes of HMP shunt, G6PD, for example. Okay, fine. So this is making NADPH or NADH. I mean to say it is using. So this is very important question. Glutamate dehydrogenase can use NAD or NADP, anything, right? It is present in liver, I told you. Then regulation, very important question. Okay, so in the regulation part, the low energy. First, I'm writing direct points. ADP, especially this ADP, you have to remember, and or the GDP, right? They are the allosteric activator. So you have already read a lot of things about allosteric regulation. Okay, so allosteric regulation. So which are binding at a different site than active site and regulate the activity of enzyme. Just change the Km and Vmax of the enzyme, right? So as ADP, and first I'm completing this topic. GTP, you have to remember most importantly, and ATP also. They are allosteric inhibitor. Okay, allosteric inhibitor. Very important question for almost every enzyme exam. They have asked this question. Okay, now let's understand why this ADP is activating this enzyme. So very simple, right? See, ADP means low energy. So what is a low energy signal? ADP, or even it can be GDP, right? So low energy signal activate the glutamate dehydrogenase. Why? Right? Because this will generate the alpha ketoglutarate. So what? This alpha ketoglutarate enters the Krebs cycle or the TCA cycle, right? And there obviously it will generate huge quantities of ATP or energy. Clear to all? So this is the purpose. Fine. So in the low energy state, this pathway will get activated in the early stages, right? Okay, like for first 48 to 72 hours, the metabolic pathways change a lot, right? After 48 hours, 72 hours, what happens? Actually, the ketone bodies take over the major metabolism in our body, right? Okay, but before that, there are a lot of things happening: gluconeogenesis, glycogenolysis, right? This process, it is unnecessarily generating ammonia, right? Okay, our muscle proteins are broken down in the fasting states. Okay, that's why fasting is not should not be continued beyond 14 to 16 hours, right? Because that time the gluconeogenesis is at peak, and gluconeogenesis, when it's peak, it is breaking the muscle amino acids, and amino acids convert into glutamate. Glutamate can generate ammonia also, but that is not toxic because our body has the ability to handle it. Okay, so that toxicity happens only in cases of liver failure, right? Mostly when the urea cannot be made. Am I clear, guys? Everyone clear? Why it is happening so? Okay, so that's all about glutamate dehydrogenase, a very important topic. Okay, so this is oxidative deamination. Fine. Now next is third point is transport of ammonia. Okay, transport of the ammonia. Okay. So guys, how the ammonia is transported? Okay. So ultimately you can see ATP is, ammonia is generated. Okay. So guys, what happens? Right. So unnecessary high amount of protein consumption is of no use. Right. Okay. Sometimes it happens like people. You must have seen that people go to the gyms or the health clubs. They do nothing there. They just go there. They watch people not doing nothing, no exercise, right? But what they do, they start taking diet. For what? Because they are going to gym, but they are doing nothing. Okay? They are buying protein, right? Worth thousands of rupees. Okay, and what happens ultimately to that? Now all these thousands of rupees are converted into urea and appear in urine, and that is causing unnecessary load to the liver, right? Generating acids from the proteins, which are causing precipitation of calcium in the kidney and causing stones. I mean to say, this is also a form of malnutrition, taking excess, right? And that is not at all desirable, isn't it? Okay. Oxidative deamination occurs in mitochondria. Kabir Singh. Okay. So transport of ammonia. Next. So as ammonia is toxic. Right. So ammonia is generated in various metabolic reactions. Right, ammonia is generated in the various metabolic reactions, right, and also produced in large quantities, okay, by the gut microbes. Okay, so the microbes or the bacteria which are present in our intestine, okay, so they are producing the large quantities of ammonia every day. Fine. Okay, that's why you will read, or you must have read, or probably you will read later in your medicine that in your pharmacology you must have read that you give some antibiotics in a patient of hepatic encephalopathy to kill the microbes present in gut because they are producing excess ammonia, which is causing problems. Okay, because of ammonia generation. Okay, so this is physiological. Okay, so this ammonia enters or easily crosses the blood-brain barrier. Okay, blood-brain barrier. Fine. And there, in the brain, the alpha-ketoglutarate plus ammonia, they produce the glutamate. Fine. And then one more ammonia is added here, and this enzyme is known as glutamine synthetase. Okay, and it is converted to glutamine. First try, then we'll understand it again. Okay. So whatever the ammonia is produced, okay, it can easily cross the blood-brain barrier, but it is physiological. It is not in toxic or concentration, right? So it is converted finally to the glutamine. Okay. So glutamine is the most important transport mechanism of ammonia in the non-toxic form. Okay, so question was like this: which of the following is the non-toxic form of ammonia transport? Your answer is glutamine. So where we are transporting, right? Like one more question in the same way we can say, like how the brain detoxifies ammonia. So question is how the brain detoxifies ammonia. So what is your answer? Glutamine. Okay, so answer is glutamine. Clear? So the two ways to ask the same question. Okay. Fine. So this is physiological, right? Normal conditions. Every day what we are doing. Brain is very sensitive for ammonia. I'll tell you why. Okay. So this is how we transport. Now this glutamine, we can say. So we had this glutamine, and this glutamine transports to liver. Okay. So glutamine enters the liver cell, okay, or the kidney cells, right? So in the liver cell, this glutamine is again acted upon by the liver glutaminase. Okay, so what is the name of enzyme? Glutaminase. Fine. And which releases ammonia, produces glutamate. Fine. Glutamate. And this is converted to alpha ketoglutarate again by the same enzyme, glutamate dehydrogenase, and it is again releasing ammonia. Fine. And guys, from this ammonia what we get is urea. So you can see from any tissue, right, we can say that this is blood-brain barrier. I am talking about brain, but this can be any tissue, right, or any tissue. So from most of the tissues in our body, right, the ammonia has been converted to glutamine, and now it is reaching back to the liver. It should be there. Okay, so it's not just brain. Brain I have just mentioned because it is the most important question and most important organ affected by high ammonia levels. Not other tissues are affected much. Okay, so that's why. So I mean to say this is any tissue, you can see. Okay, guys. So now the alpha ketoglutarate is formed, and this can be used for any purpose, like in the Krebs cycle, for example. Right, so this can be used in the TCA cycle to produce some energy. Right, guys? Okay, guys. One more important concept that I want to tell you here that you must be knowing already: kidney. Right? What is the role of glutamine in the kidney? So, kidney is not producing any urea, first of all. Fine, but kidney has to maintain, in the conditions of metabolic acidosis, right, where the acids or protons are high. Fine. So this glutamine is converted into glutamate and releases the ammonia, which is very important for kidney this time. Okay, as you know why? Because this ammonia, so ammonia binds the acid and excrete it. So this is the compensation for metabolic acidosis. Is it clear to all? Right? So the kidney glutaminase is not expressed much normally, but if we consume, if the dietary changes are there, which are more acidic diet we are taking, or there is a kind of acidosis present in the in our system, which may be even mild, then this glutaminase enzyme will be activated. So this glutaminase will also be acting here. It is also acting here. Everyone clear? Very important concept and MCQ. Okay. And that is fine. That's all about the excrete in the urine. So guys, that's all about ammonia transport. Now the question is, the MCQ is, so why the ammonia is toxic in high concentration? In our blood, obviously, right? Not in test tube. Okay, so why the ammonia is toxic in the high concentration? Fine. So the reason for that is answer. Direct question, highly repeated. Okay. So guys, ammonia, I told you, enters the brain and bind alpha ketoglutarate, I told you. So this way it will deplete the alpha ketoglutarate of the brain Krebs cycle. The most important pathway in our cells which is generating energy is Krebs cycle, as you know, right? Okay. Krebs cycle is the center of all metabolism. Whatever you consume, carbs, lipids, proteins, everything ultimately enters into the Krebs cycle in the various forms. Okay, like pyruvate can convert into acetyl-CoA, lipids can form acetyl-CoA, proteins, amino acids can form different compounds like succinyl-CoA, alpha-Kg, fumarate, and they can enter again into Krebs cycle. I'll tell you how. So, deplete. This is the point that you must remember. Okay, everyone. So deplete the alpha ketoglutarate of brain TCA cycle, and this leads to brain dysfunction. Okay, so this will cause brain or neuronal dysfunction, which may manifest as, let's say, coma, etc. Okay. altered sensorium, coma, seizures, etc., right? So it can be anything. Okay, so that is the point. As per one more theory, there is excess glutamine. Okay, excess glutamine also accumulate in the brain cells, and guys, it is causing cerebral edema with the unknown mechanism, right? Okay, so as per the recent advancements in understanding this process, this was found that brain cells of a patient died of hepatic encephalopathy or ammonia toxicity, they had very high amount of glutamine accumulate in the brain cells. Okay, and this lead to the cerebral edema. Okay, especially cerebral edema has been explained based on this. But how the glutamine is acting as something which is causing edema, right? It is not very clear to me. So this can be the new point. Okay, guys. So also remember one thing: blood ammonia levels are reflected by blood or brain, whatever it is, CSF, glutamine levels. Understand? Okay, glutamine levels. Okay, so there was a question like this that a patient presented to the emergency with altered sensorium and later on coma, right? So he had a history of aversion of protein containing foods, right? Okay, and he had also history of, he was alcoholic and he had liver cirrhosis, right? Okay, so liver cirrhosis, and he is a, let's say, the known, he has already previously known case of liver failure, right? Progressive liver failure. So which of the following amino acid may be present in very high concentration in the blood? So progressive liver failure, right, and altered sensorium, coma, right, these all are going to one point, and that is defects in urea cycle, or I mean to say defects in liver making urea. Fine. So that will ultimately lead to high ammonia level, but they asked amino acid, so your answer will be glutamine. Are you clear, everyone? So when you say ammonia, always keep in your mind glutamine, right? If I say high ammonia in this patient, I can also say high glutamine in this patient, right? Because that is causing all the problems of making glutamine by using alpha kg. Hope this point is very clear to you. Next is, so now we'll discuss urea cycle that you know. So this biochemistry is not all about cycles, like cycles are pointless, isn't it? I mean to say, there are just two cycles in biochemistry. Just one is a Krebs cycle and one is a urea cycle, the two important cycles. And these two important cycles must be your plus point in exams. I mean to say, you should be very well versed with these two topics, Krebs cycle and urea cycle. Metabolic acidosis. I was telling you, Tanish, I was just telling you that kidney also has, glutamine can enter into liver or the kidney. But in kidney, the glutaminase is not active all the time. It activates only in the conditions of acidosis, metabolic acidosis, and that time the ammonia which is released will help in removal of acids from the blood. Okay, so compensation of metabolic acidosis. Kidney and lungs are best friend, right, while handling alkalosis and acidosis, right? That is your medicine part. But this basic point that you must know, I explained it. Okay. Clear? If clear, say okay. Okay, so urea cycle, very important topic. I mean to say everything we are going through is urea cycle only, just cyclic steps now. So for the site, it is only liver, please, not kidney. Kidney doesn't make urea, right? Kidney excrete urea. So liver, in the liver, both mitochondria plus cytoplasm. So first two steps in mitochondria, right? So liver, mitochondria plus the cytoplasm. Okay. And then our purpose, what is the purpose? To detoxify the ammonia, right? So what is the purpose? To detoxify ammonia as the non-toxic urea. What is urea? NH2CO2. So that means it has total of 2 nitrogens. Okay, so in one urea molecule every time we are removing 2 nitrogen in the urine, right? So urea cycle. Fine. So let's start with steps. So guys, it is a cyclical process. I am just taking it in a linear form, and rest what is generated like ornithine will go back to the mitochondria. So let's start with mitochondria. Pointers we should remember. This is the only cycle except the Krebs cycle. These two cycles you must remember always. Not just mug up. You have to understand and remember the concepts. Favorite of examiners till now, right? Exam pattern may change, but examiners gonna same, right? They will not leave their job, right? Okay. So mitochondria, fine. So mitochondria is so first of all, guys, we have ammonia, right? So ammonia is generated. I told you how. Then we take CO2. So you know the product formed, carbamoyl phosphate. Okay, so this is CP in the short, so the name of enzyme is CPS1. Okay, as CPS1 is a rate limiting step in this pathway. Fine. CPS1 is the rate limiting step. I'll explain later, but just write that NAG, N-acetyl glutamate, is the most important activator of this enzyme. Okay, most important activator of this enzyme, NAG, N-acetyl glutamate. Just write this. I will explain everything and will write again. Carbamoyl phosphate is then added with amino acid which is generated from arginine, ornithine. Okay, so ornithine, name of enzyme in short is OTC, ornithine trans carbamoylase. Simple name. Transfer the ornithine to carbamoyl phosphate. Okay, so guys, what we get is known as the citrulline. Okay, so this we started in mitochondria. Now this will be in the cytoplasm. So guys, only first two steps, right? Only the first two steps are in the mitochondria. Rest all the steps are in the cytoplasm. Fine. So this citrulline will be sent out with the help of the special transporter known as ORNT1. Just write short form ORNT1, ornithine translocase. I'll write it. So citrulline is out in the cytoplasm, and citrulline is then added with aspartate. Okay, so we will take which amino acid? Aspartate. Fine. So aspartate is added here, and then name of enzyme is name of the product form is called as arginosuccinate, in short like AS. So this enzyme is arginosuccinate synthetase. Okay, arginosuccinate synthetase. Fine guys. Okay, so this and the first reaction. So again I'm coming back to the first reaction. Guys, this uses 2 ATP. Right? Directly it is using 2 ATP. So ATP is converted to ADP. Right? 2 ATP converts into 2 ADP. Okay. And this arginosuccinate synthetase, right, is also using indirectly 2 ATPs. Actually, what happened here is ATP is converted into AMP. Fine. So we have used the 2 high energy phosphate bonds of almost equal energy, right? 2 high energy phosphate bonds. Okay, so guys, we count it as 2 ATPs. Fine. See, when the ATP converts into AMP, we are counting it as 2 ATPs. First, that you did in your activation of fatty acid. In the beta oxidation of fats, you counted as 2 ATP, no? Right? Activation of fatty acid, you counted 2 ATPs. That was also ATP to AMP. This we count as 2 ATPs, right? ATP to AMP. tRNA activation, that is attachment of amino acid to the tRNA. ATP to AMP, we count it as 2 ATPs. So there is a lot of controversies created in the books, like they have counted it as 1 ATP. No, yeah, this is also 2 ATPs, right? And most of the standard books, it is 2 ATPs counted. In the same way, the others are counted 2 ATPs. So guys, how many ATPs are used in urea cycle? 4. So remember, the best answer will be 4 ATPs if given in the option. Otherwise, 3 ATP, 4 high energy bonds can also be the answer. Fine. So arginosuccinate. Now guys, arginosuccinate lyase, phosphoenzyme. So arginosuccinate lyase is the enzyme which produces the amino acid arginine. Okay. So lyase, when it is lyase, it is producing arginine and it is producing the fumarate also. Right guys, fumarate. Guys, this fumarate can actually enter into the Krebs cycle. Right? In the Krebs cycle, it can convert into oxaloacetate, and oxaloacetate with the help can convert into aspartate. Fine. Okay, and guys, this aspartate can be utilized here. Are you clear? See again. So guys, the name of enzyme is arginosuccinate lyase. Okay. Sorry. This produces fumarate. Okay. Fumarate is entering to Krebs cycle, making oxaloacetate as a final product, which using AST can make aspartate again, and aspartate can be utilized here. Fine. So we can say fumarate and aspartate are links of the Krebs cycle with urea cycle. Fine. The last point is arginine is acted upon by the enzyme that's called as arginase. Okay. So this generates the urea and plus ornithine. So guys, this ornithine will be recycled to the mitochondria via ORNT1. Okay, via ORNT1. Clear to all? So this is nothing but just a simple cyclical process. So ornithine, you can make it like this. This ornithine is going back, okay, through this ORNT1, okay, and will be added here. Fine, that's all about the reactions of urea cycle. Let's take the MCQs which are asked in exams most commonly, right? Rest you will practice. So the first question is, what are the source of the 2 nitrogens in the urea? And your answer is aspartate and ammonia. Okay, aspartate and the ammonia. These guys never mark arginine. Arginine is not amino acid which is added. It is itself derived from ammonia and aspartate. Clear? So never mark arginine. Very common question. These all are important MCQs till now asked in exams and repeated number of times. Okay. Number 2. Number 2 is, I told you what is the rate limiting step. Okay, before that, what is the length of cycle with the TCA cycle? And your answer is 28 or aspartate. And if you have to ask this question, if you have to mark one answer, right, then aspartate. And this is final. No one will ask again, no one will tag any faculty, right, that what should I mark, here it is given this, there it is given that. Okay, so even if in the DQB probably there is one point which is given in a different way, so you mark this as answer, final. Okay, this was also the recent NEET exam question. So aspartate, and sometimes the question will be like this: which of the following amino acid is a link of the urea cycle with the Krebs cycle? Answer is aspartate, because fumarate is not amino acid. Aspartate is amino acid. Okay. Number 3. So how many ATP is used? Best answer is 4 ATP final, or you can say 3 ATP, 4 phosphate bonds, high energy phosphate bonds. Okay. High energy phosphate bonds. Right? Question number 4th. Okay. Question number 4th is regulation. Okay. So I told you CPS1 is the rate limiting step. Fine. The activator of CPS1 is an acetyl glutamate, that is NAG. Okay, very important question. Find how it is made. We take acetyl CoA plus glutamate in the liver. Okay, and this enzyme is called as synthase. Fine. So what is the reaction that we are taking? Acetyl CoA plus glutamate. Name of enzyme is NAG synthase, and this produces NAG. Fine. Most important allosterically activator. Only one. right? And guys, this energy synthase is induced whenever we consume a high protein. So that is what I told you. People start taking proteins unnecessarily without exercising, suppose. So what happens whenever anybody consumes high protein diet for more than 4 days, 3 to 4 days, right? And especially if this also contains arginine, right? And guys, this arginine will activate this enzyme. Which amino acid? So this is also the MCQ. Okay, and this is one of the therapy, right, of the hepatic or the disorders of urea cycle, right? Arginine, a child who has been given arginine, can make more urea and detoxifies more ammonia. Reason this arginine is the very important therapy in case of urea cycle disorders. Not all, but yes, initial few reactions are important, right? Okay, guys. So that's all about regulation. Simple. Okay, so that's your question number 4. Question number 5. Urea cycle disorders, right? Urea cycle disorders. Okay, so guys, urea cycle disorder, right, will definitely be very important, okay, in the upcoming sessions of exams, which are based on more on the clinical pattern of questions, right? But again, I told you points will be same, nothing new, right? Okay. Okay. So urea cycle disorders. Number 1. Type 1 hyperammonemia. Type 1 hyperammonemia. Species of which enzyme? Right? Okay. Most okay, with very high amount of ammonia, or you can say very high amount of glutamine. Glutamine. Question was in the All India exam like this recently, 1 or 2 years back. The question was like this: which of the following urea cycle disorder will have the maximum inhibition of glutamine in the blood? Answer is type 1 hyperammonemia or CPS1 deficiency. Clear? Obviously, here the first enzyme is deficient. Ammonia is not at all bind with anything like CO2 or anything. Clear? So that's your point. Maximum elevation. So type 1 hyperammonemia, right? And the death okay, so this patient will present with especially conversions, right? Okay, conversions. Why the conversions are there? Due to cerebral edema. Okay, this patient presents with colic not metabolic, respiratory. Okay. Respiratory alkalosis. Why? Hyperventilation because of brain damage. Okay. Why there is respiratory alkalosis? You can write hyperventilation. Okay, because of the brain damage. Okay. There will be delayed milestones, but this child will not survive much. Okay, so delayed growth, if in case survives, deficiency is not very severe, right? And there is aversion to protein food. So guys, this is like for all the urea cycle disorders. Okay, definitely this will be the most severe case. Okay, so most toxic, and here death occurs before 5 to 10 days. Okay, death by 5 to 10 days. So there are no chance of delayed growth or something else like that. Okay. Guys, mental retardation is very mild or nil. Okay, these are the points which will help you in the answers. So as per the various books of medicine and genetics, there is a very mild, mental retardation. And some books mention directly no mental retardation, right? Okay, so you follow this when writing. So guys, that's all. So guys, this is for all urea cycle disorders. Okay, not just the CPS one. But these clinical features, will apply to most of the disorders. Okay, most of urea cycle disorders. Am I clear, everyone? Voice break. Nothing, I'm just explaining you the clinical features, which are convergence due to cerebral edema, respiratory alkalosis, which is again because of the brain damage, hyperventilation because of the brain damage, and delayed growth with aversion to the protein containing food. This is applying to all the urea cycle disorders. Mental retardation may or may not be present, and if it is present, it is mild. Fine. Okay. For specifically CPS1, it is most toxic, highest elevation of ammonia in the glutamine levels. Is it fine? Okay. So that's all about the CPS1. So number 2, CPS2. I'm sorry. Number 2 is hyperammonemia type 2. Okay. Hyperammonemia. Okay. So division is OTC, right? So guys, this is X recessive. Fine. Most common cycle disorder. Okay, most common urea cycle disorder. Fine. Okay. So X-linked recessive, most common urea cycle disorder it is. Fine. And there is also high levels of ammonia or patient has increased uracil or orotic acid blood or urine. Okay, blood or the urine. Now this point I'll explain. Uracil or orotic acid in the urine. So that's called as acid urea. Fine. Orotic acid urea. Fine guys. So I'm explaining this now. Orotic acid urea. On the next page. Fine. So orotic acid urea. So guys, orotic acid urea is normally a disease of pyrimidine metabolism, but why it is happening in the urea cycle disorder, right? Okay. So in this case, what happens is, in the orotic acid urea, what happens is, there is a deficient OTC enzyme. So there is increased in the carbamoyl phosphate, which is in mitochondria, and this will overflow to cytoplasm, right? And this carbamoyl phosphate then enters to the pyrimidine synthesis, uracil synthesis, uracil cytosine. First pyrimidine that we synthesize is uracil. So enters to pyrimidine synthesis. Okay, and bypassing, because we'll see later on, the rate limiting enzyme of pyrimidine synthesis is CPS2. So guys, carbamoyl phosphate is also made in pyrimidine synthesis. Carbamoyl phosphate is also made in urea cycle. Name of enzymes are different, 1 and 2. Okay, so carbamoyl phosphate enters to pyrimidine synthesis, right? And then this will generate orotic acid, right, which is nothing but a pyrimidine precursor, right, or uracil, which is rare. Less amount of uracil. Why? Because you have to make the nucleotide synthesis. We also need sugar plus phosphate, which is not available in excess. Okay, obviously this is something bypassing the pathway. Carbamoyl phosphate, it is unnecessarily making pyrimidines, but pyrimidines will not be made much because ribose 5-phosphate from the HMP shunt is not much available. Okay, there is no demand. Fine. Okay, because that's a controlled pathway. But orotic acid will be made, right, like this. So this is the MCQ which has been asked in various exams. Okay, so you will get a question like a newborn child, let's say 6 months old child, has aversion to the protein foods or had episodes of seizures or convulsions at birth, etc., etc., right? The growth not achieving milestones. On analysis, his blood contains high amount of glutamine or the ammonia, and uracil is also present in urine or the blood. So when you get this point, uracil in the blood, your answer is OTC. Okay. CPS2 is acting pyrimidine, yes. Forget CPS2 right now. Pyrimidine synthesis, no? CPS2 is acting in pyrimidine synthesis. Okay. Fine. Please listen to me also, I request. No? Okay, so I'm just I just mentioned CPS2 is used in pyrimidine synthesis, and this pathway of overflow of carbamyl phosphate, right? See, again I'm repeating, orotic acid urea. So when the second enzyme of urea cycle is deficient, the first product carbamyl phosphate accumulates. Okay, this will overflow to cytoplasm from mitochondria, and in the mitochondria it will make pyrimidines. And pyrimidines, one of the pyrimidine is orotic acid. That you don't know, you will learn. If you know, that's very good. And also some less amount of uracil. So why the less amount of uracil? Because sugars are not available to make UNP. Okay, so orotic acid, they will stop at orotic acid. Okay, fine. Is it clear, everyone? Okay, so that's your CPS2. Number 3 is known as citrullinemia. Deficient enzyme is arginosuccinate synthetase. Okay, so there will be high level of citrulline. Okay, increased amount of citrulline. Fine. Number 4 is called as simply arginosuccinate acid urea. So the name of enzyme is arginosuccinate lyase, which is deficient. Okay, fine. Arginosuccinate acid urea. So they also have a mild elevation of ammonia levels, right? There is definitely high ammonia levels. There is definitely high ammonia levels, which is mild. Okay, fine. Number 5 is hyperargininemia. Okay, hyperargininemia. Okay, so hyperargininemia is due to deficient which enzyme? Deficiency of the last step, that is arginase. Okay, last step, that is arginase. Okay, arginase is water soluble, right? And soluble and can excrete in urine, but it can excrete in urine. Okay, very mild ammonia elevation, so it is not toxic. Fine. Patient present by 3 to 5 years, so it goes unnoticed. Okay, by 3 to 5 years of age with neurological complications. Okay, and one of them is like spasticity. Okay, it is very rare, most rare type of urea cycle disorder, mostly go unnoticed. Okay, and even if it is diagnosed, it is very difficult to diagnose, right? It needs a very good suspicion plus very well equipped labs to test for the enzymes, genes, etc. Okay, so it may present with spastic diplegia. Find one of the features. Okay guys, so very rare type of disorder, hyperargininemia, and it is very less toxic. Fine. Patient presents late. Okay, right, so that's your number 5. Number 6 is triple H syndrome. Okay, triple H syndrome. So guys, what is triple H syndrome? It is due to deficient ORNT1. Deficient ORNT1, which is also called as ornithine permease or translocase. Okay, ornithine permease or ornithine translocase. What is triple H? See, H for ornithine will not go back now, right, from the cytoplasm. So ornithine will go into the blood, right? So hyper ornithinemia is the one. Then H for, so hyperornithinemia. H for, definitely if ornithine is not going back, ammonia level will start rising, because this is a very important part of the second step of urea cycle now. So ammonia utilization will start decreasing. So hyperammonemia. And the third H is, okay, so carbonyl phosphate will keep on making, this is first of all, right, how homo, not hyper, homocitrullinemia, right? What do you call this? Homocitrullinemia. No, arginine doesn't appear in the urine normally. Excess arginine, if it accumulates, it can appear. So it is accumulating in this way, no? Right? Because it is not converted into urea. So the liver will, remove the arginine to the blood. Okay, and that will be in excess every day. So that may be removed in the urine. Fine. Okay. Urine may contains, this arginine contains guanido compounds. Right? Okay. So if you remember in your first year, right, you must have done a reaction a test which is known as Sakaguchi test. Sakaguchi. Okay, so there was a test this was a test which is known as Sakaguchi reaction or test. Okay, what it was for? It was for the guanido group. Guanido, okay, group of arginine. Right, you can get this test positive. Right? Fine. Sakaguchi test. Fine. Okay guys, remember this is not hypercitrullinemia, homocitrullinemia. What is homocitrulline? It is carbonyl phosphate plus lysine. Okay, lysine of proteins. So actually what happens, the excess carbonyl phosphate which accumulates, okay, because urea cycle is now stopped first at the step 2 now. So but the carbonyl phosphate was still made, it binds with lysine and makes homocitrulline. So that's, you can ignore it, just remember the three names: hyperornithinemia, hyperammonemia and homocitrullinemia. Right guys, so these are the three important, six important urea cycle disorders. Right? So as like how can you answer an MCQ? So you have a newborn child. Okay. So in the newborn child, there are features, various features. Okay, I'll show you one. Picture I'll show you. Basically, you can answer the question of urea cycle disorder based on this uracil or the orotic acid is raised or not. See, this is a newborn child. Okay, so newborn child which presents with convulsions. Okay, so when the newborn child presents with convulsions or acidosis, okay, convulsions, fine. So you can look for two things, right? If it is acidosis also present, right? So if acidosis are present, then it is not a urea cycle disorder. Okay, not a urea cycle disorder. Fine. If you find respiratory alkalosis, right, plus high amount of ammonia levels, then you will suspect the urea cycle disorder and urea cycle disorder. Now, if urea cycle disorder, you suspect urea cycle disorder, first of all you check if there is increase in amount of the orotic acid, right? So if increased amount of orotic acid is present, right, then what is this case? Deficient ornithine transcarbamylase. And if it is not, then this can be a case of, most importantly, CPS1, because a child is so young, less than 24 hours of age, right? Child, let's say 24 hours, because other disorders will not present so early. Okay, so if orotic acid is not raised, it is CPS1. If it is raised, it is OTC. Then you can look for one more thing. Fine, if it is not conclusive, is there any increase in citrulline levels? Okay, if there is increase in citrulline levels, right, then this is the deficient arginosuccinate synthetase. Okay, arginosuccinate synthetase. Fine. Everyone clear? So with this way you can identify. So only 2 points if you have, orotic acid and citrulline. So again listen, child with some clinical features and hyperammonemia. If he has high orotic acid, this is the answer. If he has high citrulline plus little amount of orotic acid, it is arginosuccinate synthetase. And child has nothing elevated except ammonia, type 1 hyperammonemia. Am I clear? Child has nothing at all and nothing else is elevated except the ammonia or glutamine, it's type 1. Ammonia plus orotic acid, OTC deficiency. Ammonia plus little amount of orotic acid plus high amount of citrulline, arginosuccinate synthetase. Okay? Everyone clear? And arginosuccinic acid in the blood and arginosuccinate lies, that is easy. Arginine is very high, so arginine is deficient. Am I clear? So that covers your urea cycle. In the treatment part, I told you can give arginine. Okay? Because arginine has two roles. It provides ornithine. Okay? It provides ornithine for OTC reaction. Second, it activates NAG, which is the activator of rate limiting enzyme CPS1. So why we should provide arginine in the diet of a patient of urea cycle disorder? Answer is this. Second is definitely low protein diet. Okay, obviously you know why, right? So low protein diet. Fine. Number 3. But calories must be maintained. Why? Because otherwise the muscle will be broken down. Gluconeogenesis. You have to maintain the calories. So low protein diet, which is rich in carbohydrate. So carbohydrate or sugar should be there for the baby to survive. Okay, carbohydrate rich diet. Third, there are some drugs. Okay, there are some drugs. Number first is phenylbutyrate. Okay, phenylbutyrate. Okay, phenylbutyrate is a nitrogen scavenger, or you can say the drug that I'm writing these are nitrogen scavengers, right? So these are very important MCQs. For example, this phenylbutyrate, okay, it is taken orally and it's a prodrug. Fine, it is converted into the active drug, phenylacetate. Okay, so that's the active drug. This phenylacetate, PA. Okay, so PA. Okay, PA phenylacetate binds the which amino acid? Glutamin, which has 2 nitrogen and which was already very high in the urea cycle disorders. Okay, and this produces the compound that's called as phenylacetyl glutamin. Okay, phenylacetyl glutamin. Clear? So phenylacetyl glutamin is non-toxic, soluble, water-soluble compound excreted in the urine. So this will appear in the urine. So 2 nitrogens excreted. Okay guys, so guys, these are called as scavengers. Okay, nitrogen scavengers. Clear to all? Everyone? Okay. So this is one of the very important MCQ in a drug, right? That you should know. Phenylbutyrate or phenylacetate. Right, repeated number of times in exams, including this year. I mean to say, yes, this year, right? So, what is the other drug? Other scavenger is the benzoic acid, which is a common additive to a lot of drugs and foods. Benzoic acid. Why? Because benzoic acid binds with non-essential amino acid glycine. Okay, and this is converted into a water-soluble hippuric acid. So 1 nitrogen excreted. 1 nitrogen excreted in the urine. Okay, guys. So that's all. But rest you will read a lot of things in your medicine also, and that is like you have to give some antibiotics you can add. Right? What antibiotic you should add? Rifaximin. Right? Why? Antibiotic rifaximin kills the gut microbes and increases ammonia levels. Right? And you can also use hemodialysis. Okay, to all. That's it. A very important topic. Right? Everyone clear? So that's all about the urea cycle. Now we'll start with our next topic. Okay. flowchart is nothing here. Flowchart is very simple. See, the flowchart is like this, that when a child presents with convulsions, right, and age is around 24 hours, okay, so when the child has acidosis also apart from convulsions, right, so that's not a case of urea cycle disorder. Urea cycle disorder doesn't present with any acidosis, okay. So you can check for respiratory alkalosis or high ammonia levels, okay. Acidosis may be present in other conditions like beta oxidation defects and some other amino acid ureas that I will discuss now, right. So simply remember like this: you have a case of a child presenting with some clinical feature in high ammonia levels. If high ammonia levels, then you will suspect urea cycle disorders. Then you will check orotic acid. If orotic acid is not high, then it is type 1 hyperammonemia, and it is high, then it is type 2 hyperammonemia, right. Then if orotic acid is having no high levels, you will also check for citrulline levels. So if citrulline levels are very high, then you will mark the answer as arginine synthase defect. Am I clear? Fine, guys. Okay, so this is your arginosuccinate synthase. Okay, guys, so that's all about the urea cycle disorders. Sorry. Break. Okay, we'll take a small break. Fine. Come back in 10 minutes. Right, I'll start with our next topic. Fine. Okay. So guys, what we need to do is, like, we should start the things that we need to read or revise. Okay, because the biggest problem is we don't start, and especially the subjects. If you find them a little difficult or tricky, then you avoid them. So it's better always to start revision. Okay, doing MCQs. Right, take any one topic and start. Just don't wait. I will start from page 1 to page last. That's sometimes not possible. Right? It's always to go ahead. It's better to go ahead and start at least one topic that you feel is easy for you, right? And you can cover it up easily. Okay, so our next topic is amino acid metabolism. Okay, so now we will talk about the disorders and special products of amino acids. Okay, so write the heading: next chapter of the same amino acids. So disorders and special products of amino acids. So we'll take one by one. Thank you, Mansi. Okay, so I'm talking about disorders, right, and products. So we'll take one by one important amino acids, not all 20. Don't worry. So we'll take some important amino acids and their disorders which are important for MCQs, right? So let's start. Number 1. So number 1, I'm starting with the sulfur containing methionine. So guys, methionine has sulfur, not as such. This was a question asked. Okay, sulfide groups are present in cysteine, not the methionine, right? Okay, so not the methionine. So guys, methionine is a sulfur containing amino acid, essential amino acid, as you all know, very essential amino acid. Okay, and So let's start with their basic metabolism. So guys, methionine using the ATP, okay, with the help of enzyme MAT. MAT is methionine adenylyl transferase. Okay, methionine adenylyl transferase. Abbas is also listening to my lecture. Kalbe Abbas. So methionine adenylyl transferase, that is MAT, and then this is converted into sulfur form transfer S adenylyl methionine SAM. Okay, SAM, SAM. So on the sulfur we have transferred the adenylyl group. So we'll take the importance of everything later. First I am completing the flow chart of the basic reaction. Okay, SAM is a methyl donor that I'll tell you in details. Okay, so it donates the methyl group and it is converted to the compound that is homocysteine, right, that you all know, homocysteine. Fine. Then this homocysteine is bad for our health, right? It is a risk factor for the CVDs. So this is added with a amino acid that's called as serine. Okay, serine is added here, and the product which is formed is known as cysteine related. Okay, homocysteine. Then we have cystathionin, cystathionin. Okay, so thiol groups are related with the S or the sulfur, right? So thionin, guys, this enzyme, if I take this C, then this enzyme, please write cystathionin synthase. Okay, cystathionin synthase. Am I clear? Then this enzyme cystathionin synthase, okay, it is a very, very important enzyme, and it is dependent on the vitamin B6. I told you, now, if you don't know anything and there is an enzyme of metabolic pathway of amino acid, you can at least take a risk which will be low risk to answer B6. So this is vitamin B6 dependent. Cystathionin is then acted upon by various steps. Okay, so no need to remember them, and what we get is ultimately cysteine. Okay, cysteine, so which has as such groups here, right? So we will remove, because a lot of pathways are there, a lot of reactions are there. What I'm telling you, you must remember or try to remember them. Okay, at least remember all the MCQs, practice them, and you'll be able to remember more. Right? So what we get is cysteine. Okay, so methionine is finally converted into cysteine. So cysteine is a non-essential amino acid. Okay, now let's talk about MCQs. Okay, one by one. Right. Number first is deficiency of methionine adenylyl transferase. Right. This is called as hypermethioninemia. Fine. Now, guys, this condition has no particular clinical picture, except just you have to remember one thing, right? And that is, this patient presents with boiled cabbage order. Okay, boiled cabbage type of. Guys, these are important questions that we will cover 4 to 5 types of orders. Okay, typical orders in amino acidemias, amino acidurias. Okay, and these are asked as MCQs. Okay, and these are hints to the questions, very easy hints that we can answer. So boiled cabbage body order. Okay, so hypermethionemia. Methionine is not metabolized. Number 2. So we were talking about SAM. SAM is really very important, very important part of our biochemical system, right? SAM is also a very important question, right? So SAM is a methyl donor, right? SAM is a methyl donor, fine, and it is a methyl donor in various reactions, fine. So what are the reactions? Number 1, in converting norepinephrine to the epinephrine, just a methyl group is required, which is given by SAM. Okay, we can take like this A, B. Next is SAM is very important in converting serotonin to the melatonin, which is a hormone of pineal gland. Okay, so we will take again them in some other pathways, right? Serotonin to melatonin. Okay, so that's the second one important point about SAM. Okay, so SAM is required for various other reactions like DNA methylation reactions. So methyl donor, right, which can silence the genes, methylation. Okay, regulate the genes. Interesting, right? Here is, so that's your DNA methylation. So as these two are very important, especially serotonin, melatonin. Okay, number 3. Number 3 disorder is known as homocystinemia, or also called as homocystinuria, right? So in this disorder, what is the cause of this disorder? The most common cause is deficiency of the enzyme cystathionin synthase. Okay, so I mentioned this already, cystathionin synthase. Fine. So most common cause is cystathionin synthase, deficiency of this enzyme. Right, again a very important question. Right. Then there is increase in the levels of homocysteine, HOC I'm writing, right? HOC, homocysteine, and there's this homocysteine, okay, high elevated homocysteine levels. This is the risk factor, independent risk factor for cardiovascular diseases. Very important question. Okay, risk factor for CVD. Actually, why this homocysteine is, this homocysteine is converted to, high amount of homocysteine is converted to the homocysteine thiolactone, which is a reactive compound. Okay, thiolactone. Thio means disulfide bond. So homocysteine thiolactone, right, which react with LDL, the bad cholesterol, low density cholesterol. Okay, this will lead to the LDL aggregation. Okay, and these are, as you all know already, taken up by the macrophage. So guys, बच्चों सबको याद है ना ये point? Everyone remember, kids? You remember why LDL is bad cholesterol? This is the reason, right? Because LDL is actually, excess LDL is taken up, oxidized LDL is taken up by the macrophages, oxidized. This is also oxidation. Okay, so uptake by macrophages, and then what happens? You all know, foam cell formation, and this leads to risk of CVD or atherosclerosis. Okay, so this has not been asked. The question is only till now that which amino acid is a risk factor for CVD. So one-liner or even clinically asked, and this is the answer. Okay. So these are mostly young patient. Okay, young patient. So nowadays, homocysteine test is a common test used by doctors, right? So it was previously very costly, but nowadays not very costly, and it is easily available test. Okay. So this is what happens. That is okay. One more point. The patient has clinical features resembling the Marfan syndrome. Right? This occurs if the case is a child, the early case. See, homocysteine levels may rise late in the life, adult life. This is a child. You know, patient has a clinical feature of Marfan syndrome, right? That's also called as Marfanoid features. So if you see this term, Marfanoid features are seen in homocysteinemia. Why? As the homocysteine bind the gene fibrillin protein, right, which is defective in Marfan syndrome also, right? Okay, so homocysteine binds the fibrillin. That's why. Okay, so there are some features which can differentiate. See, all the clinical features of Marfan syndrome, like arachnodactyly, tall and thin limbs. Okay, they are all present. Pactus carinatum, pactus excavatum. These are which are present in Marfan syndrome also, right? So actually, high homocysteine and Marfan syndrome, right? So they may differ into 2 points. Definitely the clinical points, okay, because the deficient enzymes or the deficient proteins you already know. So in this case, there is lens dislocation, which is also seen in Marfan, but in this case it is temporal, upward and outward, right? Okay, lens dislocation is upward and outward. Here it is inferonasal, downwards and inwards. Very important point 1. Second point is the joints. Okay, so in this case you all know these are legs, okay, or hypermobile joints. Fine, but here they are stiff, clearly differentiated from Marfan syndrome. So even arachnodactyly or the spider-like fingers or tall limbs or whatever the features are same in both the cases, but these 2 important features are very important. Definitely lab diagnosis, high homocysteine levels can be there, right? Okay, so everyone clear? So joints are stiff, and here there are hypermobile, as you must be knowing already, right? Marfan syndrome. At least those who have covered pathology must be knowing it very clearly. There guys, so this is the difference. That's it. Okay. So guys, what is the therapy you can give? Try 5 doses of vitamin B6 or B6 plus B12 plus why these two other vitamins? Why B6? Everyone clear? Why B6? Okay, because B6 is the activator of cystathionin synthase, right? That's why you have to give B6. But why B12 and folic acid? Okay, let's understand this part. So, increased homocysteine, hyperhomocystinemia is by humans via making melatonin from homocysteine. Okay, homocysteine. So homocysteine and methionine differs only in one methyl group. So guys, if you add just a methyl group, right? For this addition, you need an enzyme that's called as methionine synthase. Okay, methionine synthase, and this is dependent on a very important vitamin, that's B12. Okay, so that's called as methyl cobalamin. Okay, so guys, this is the name of the vitamin, methylcobalamin. So this methylcobalamin converts to cobalamin. So methyl donor, here it is, methyl donor, okay, to the homocysteine, right? Now guys, this cobalamin should be converted back to the methylcobalamin, and this is done with the help of tetrahydrofolate, okay, which is converted into tetrahydrofolate or the folic acid, right? So what you can see here is that I already told you that homocysteine and methionine, they differ in methyl group, right? So first we'll convert homocysteine to SAM, then SAM to methionine. That's the same thing, reversing the pathway. We are just reversing the pathway. So now we can convert homocysteine to methionine with the help of two vitamins, B12 and folic acid, both methyl forms. Okay, both are the methyl forms. Am I clear, kids? Everyone? Fine. Okay. So what is the name of this very important enzyme? Methionine synthase. Guys, here there could B12 in our human body. There are just two roles of vitamin B12. This is the one, and one you already covered. What was that? To convert propionyl CoA generated from ornithine fatty acids to the methylmalonyl CoA. Yes, succinyl CoA to the methylmalonyl CoA. Remember everyone, yes or no? Okay, there are just two roles of vitamin B12 in our cells, in our body. Okay, only two, easily remember, right? This is the second enzyme, methionine synthase. And it's so, okay, so in case of what I can say is, there is a condition that's called as non-classical homocystinemia. Okay, so this is due to deficient B12 synthesis. So how will you differentiate from classical homocystinemia? Right? Simple here. If B12 is not formed, here the B12 is not formed. Fine. So this patient has definitely increase in the homocysteine levels. Fine. But there is normal methionine levels. Okay. Just remember, that methionine levels are high. Okay, methionine levels will be high in the classical. There is a high methionine levels in the classical cystathionine synthase deficiency system numbers. You will be it will be very easy to answer for you this question if you know about the methionine levels, and definitely in this case there will be megaloblastic because we okay. Did all okay. Fine. Okay, I'm changing the page. Hope you are done with these points. Okay, megaloblastic anemia. We are talking about non-classical homocystinemia. Clear? Changing the page. Okay. Fine. Now there is one more point we can explain here, and that is called as folate trap. Okay, folate trap. So just remember that methyl folate is only used in the above reaction. Okay, now fine, now clear, voice clear everyone. Now clear? Say yes. Now clear everyone? Okay. Should I move now? Okay, so methyl folate is only used in the above reaction. Right? And released as the free tetrahydrofolate by the vitamin B12. Okay, by the vitamin B12. So in the deficiency of vitamin B12, minus means deficiency. So guys, I am now talking about folate trap. Okay, so we converted homocysteine to methionine, right? So we used two vitamins, B12 and folic acid. Which form of folic acid did we use? Methyl folate. Okay, so what we used is methyl folate, methyl tetrahydrofolate. So guys, remember, methyl tetrahydrofolate is only used in the above reaction, methionine synthase, and it is released as the free THF by the vitamin B12. So if in case vitamin B12 is deficient, okay, folic acid is trapped as the methyl folate or methyl THF. Okay, as the methyl THF, right here. Okay, and not available for other reaction, right, which need THF free. Okay, right guys. So because of this, like for example, purine and pyrimidine synthesis that I will take in the purine pyrimidine section, how it is helpful, right? So because of this, decreased in the DNA synthesis and guys, this leads to anemia, and that is called as megaloblastic anemia. Okay, megaloblastic anemia. Megaloblast, because the nucleus is not growing, but the cytoplasm is growing, right? So this leads to megaloblast, okay, with the small nucleus, right, and the big size of the cell. We'll talk about this more again in vitamin B12, and we will talk more about this in the pathology part. Okay, so megaloblastic anemia. Everyone here, this is called as folate trap. Okay, folate trap. Voice not clear still? Now clear? Okay guys, that's all about methionine and homocysteine. So what is the treatment of homocystinemia? B6, B12 and folic acid. Clear? Right. So now let's take the next topic, next question, and that is cystinuria. Please look at the spelling. Cystine, not cystine. It's not the E. It's not cystine. So do you know already the difference? Okay. See guys, first of all, cystine is the two oxidized cysteine linked by disulfide bond. Okay, fine. Shreya, normal methionine levels are non-classical because in the non-classical vitamin B12 or the folic acid may be deficient. So how will you convert homocysteine to methionine? So methionine levels will not change or will be low compared to the classical. Clear? So cysteine is what is cystine? So we have like 2 cysteine. Okay, minus, this is called as oxidation, removal of hydrogen. So we have 2 cysteine which are now linked with the disulfide bond, and this is what we call as the cystine. Fine. Okay, so our topic is cystinuria, right? Cystinuria. So in this case what happens is, in the cystinuria there is defective basic amino acid transporter, okay, in the kidneys. So for the reabsorption, okay, basic amino acid transporter. So what is the basic amino acid? Lysine, arginine, the most basic amino acid, right? So because of this, what happens? Because of this, what will happen? So there is excretion of cystine. See, we will remember like this: C-O-L-A, cola. Okay, cola in the urine. Cystine, again, please not cysteine. This was the MCQ. Question is, which of the following amino acid is not excreted in the urine of a patient suffering from cystinuria? So cysteine nahi hoga, cystine will be there. Okay, O for ornithine, L for lysine, and A for arginine. Right? So as you know, these two are the basic amino acids. This is also basic, but not any not present in any protein. Cysteine is not basic, but it is removed through this basic amino acid transporter. Okay. So cysteine is excreted. You can write here that cysteine is also excreted through the reabsorbed through the basic amino acid transporter. Am I clear, everyone? So defective basic amino acid transporter. Fine. In this patient, I'll show you. We have that image. Because in this patient there will be excretion of some crystals of cysteine in the urine, and they look like hexagons. Okay. So 2 points. Let's write. Okay. So in this patient's urine analysis, you will find first of all rotten egg order. Okay, rotten egg order like the sulfur. Right, sulfurous compounds have rotten egg order. And number 2, right, they will be present of the beautiful hexagonal crystals. Right, so hexagonal crystals of cysteine. Okay, so they may be like stones. Right, so guys, this patient presents with the clinical feature is the cysteine stones. So the features of stone, right? Okay. So pain in the back, pain in the region, line region. Okay. Pain in the flank, right region, and urine contains these features, etc. Child. So this is the case of cystinuria. Right, guys. So what you can give this patient. Right, this patient can be again given vitamin B6. Okay, because it is involved in metabolism of cysteine also. Right, that we have not covered as such directly. Right, not required, but just remember vitamin B6 is the therapy. Right, yes. There is one more disorder, and that's called as cystinosis. The other disorder, cystinosis. Okay, cystinosis is defective lysosomal membrane transporter. Okay, known as cystinosin. Okay, cystinosin, which removes the cysteine generated by the protein degradation. In the lysosome there is protein degradation, so which removes the cysteine generated by protein degradation. Okay, to cytoplasm from the lysosomes. Okay, so it is from the lysosome to the cytoplasm, right? This movement is blocked. Fine. So because of this, what happens? Because of this, what happens? The cystine accumulate. Okay. So in the various organs, okay, cystine accumulate in the various organs and damage them. Okay, so there are two important features. One is the kidney failure, okay, and plus there is cystine crystals in the cornea, needle shaped. Okay, there are needle shaped cystine crystals in the cornea. Okay, so that is cystinosis. Okay, cystinosis is a disorder which is not cystinuria, it is accumulation of cystine. Where? In the lysosomes. LYS for lysosomes of various organs. Okay? Okay, guys. Cystinosis, repeat, there is nothing important in that. I mean to say nothing difficult in that. See, cystinuria, I told you what is cystinuria. Cystinosis is accumulation of cystine where? In the lysosomes of organs, which lead to their failure, for example, kidney failure, and maybe in the cornea, which is easily visible. So for diagnosis, it is easily needle-shaped, crystals in the cornea. Right. What is the defect? Lysosomal membrane protein, transporter protein known as cystinosin. That's it. So 2-3 points you have to remember. Cystinosins, where it is accumulated, lysosomes, why? Because of transporter defect. That's it. Okay, guys, so that's all. Moving to the next topic. So we have covered methionine and cysteine and homocysteine. Very important 3 questions we have covered. Okay, now next amino acid, and that is phenylalanine. Right, phenylalanine. Okay, aromatic amino acid. So all you know, this is the aromatic amino acid. This is the essential amino acid. Right, just one step in metabolism, and that is phenylalanine is converted to tyrosine. Okay, tyrosine. Name of enzyme is phenylalanine hydroxylase. Okay, phenylalanine hydroxylase. So that's the name of the enzyme. Here, this is the enzyme which is BH4 dependent. So it will use something that's called as BH4, which is converted into BH2. So guys, what is this? So let's take some points first of all. So tyrosine, okay, decrease the requirements of phenylalanine in diet. Okay, so tyrosine, if present in diet, so guys, this is non-essential amino acid, non-essential amino acid, right? So if present in diet, it will decrease the phenylalanine requirement. Second one, okay, BH4, 4 hydrogen, so that's why tetrahydro, and B for biopterin, okay, so tetrahydrobiopterin. This is the coenzyme of, okay, this is the coenzyme of tyrosine, phenylalanine and tryptophan, all 3 aromatic amino acid hydroxylase. Okay, clear guys? Tetrahydrobiopterin is the coenzyme of all the hydroxylation reaction of aromatic amino acid. Okay, and it is also required for nitric oxide synthase. Okay, that I will tell you with arginine nanosynthase. Okay, so that's your BH4, very important question. What is BH4? And BH4 is synthesized from a nucleotide that is GTP. Okay, from the nucleotide that is called as GTP. Fine. Now let's talk about a disorder that's called as the phenylketonuria. Okay, so just a minute, we have covered the basic reaction of phenylalanine, and what we get is tyrosine. Now let's take the reactions of tyrosine first, so that we can understand some more points. Okay, so this was a metabolism of phenylalanine, and 2 metabolism will take together, tyrosine metabolism. So tyrosine metabolism. Okay, so first of all, we take Ty for tyrosine. Okay, tyrosine is a non-essential amino acid, right? And tyrosine is acted upon by the enzyme that's called as tyrosine transminase, so which is converted into a name which is very big, para hydroxy phenyl pyruvate. Okay, so in short, that is PHPP. Okay, so first we'll take the basic metabolism, then I'll take some disorders of tyrosine and PKU, that is phenylketonuria. Okay, para-hydroxyphenyl pyruvate. So all the transaminases are B6 dependent. This enzyme is then PHPP dioxygenase. You know guys, dioxygenases are the enzymes which add the whole oxygen molecule, right, O2, in the compound. So PHPP dioxygenase, and this is converted into homogentisic acid. We'll take this in alkaptonuria, HGA. Okay, so homogentisic acid oxidase is the name of enzyme, right? This is enzyme which converts homogenesic acid to fumaryl acetoacetate, FAA. In short, the last reaction is fumaryl acetoacetate hydrolase. Okay, so what we get is, as you can see, we can get fumarate plus acetoacetate. Guys, this is a mixed amino acid, no? Okay, so mixed amino acid, both glucogenic plus ketogenic. Clear? This is both glucogenic plus, so this is glucogenic. Fumarate can make glucose by the TCA cycle, convert to oxaloacetate, which enters into the gluconeogenesis, and acetoacetate is a ketone body, as you all know. Right? Okay. Clear the one? So fumaryl acetoacetate hydrolase is the last enzyme. Now let's take some more concepts here. Okay. So let's talk about disorders. So first I'm taking disorders of tyrosine. PK you will take in the last. Okay. Disorders of tyrosine metabolism. So number 1, it is tyrosinemia type 1, which is most common. What is enzyme deficient? Fumaryl acetoacetate. So last enzyme is deficient. No, who classified them? Why they took last enzyme as a type 1, right? Because there are others which are based on some previous enzymes, but this is the most important one to remember. Fumaryl acetoacetate hydrolase, the last enzyme in the pathway. Okay. So because of this, there is increased in the fumaryl acetoacetate levels, right? Which alkylate the DNA. So DNA damage. Okay, and especially this leads to hepatocellular carcinoma. See, reason has not been asked, but tyrosinemia type 1 presents with hepatocellular carcinoma, right? Liver is affected the most. Second, increase in the amount of fumaryl acetoacetate. Okay, it is entering into some metabolism pathway, and it is converted into the succinyl acetone, which is also a metabolic poison. Okay, which is a metabolic poison. Okay, so this also leads to hepatorenal failure. So 2 organs most commonly affected. There is a succinyl acetone binds to various enzymes and block them, right? Okay, it is found to be a highly toxic poison produced from the fumaryl acetoacetate, right? Okay, this is succinyl acetone, and this succinyl acetone is also the marker, a biomarker, okay, of tyrosinemia, okay, of the tyrosinemia. Okay guys, so that's your succinyl acetone. So this can be important. Till now the only point is this one which has been important, and the name of enzyme, right? Fumarate acetoacetate hydrolase in type 1 tyrosinemia. Okay, so this can be important in your question. Succinyl acetone, right, which is a marker. Okay, it's fine. So everyone clear now? Right. So now type 1. See guys, type 2 and type 3 are just important names I'm writing because they may not be important MCQs in future. Type 2 tyrosinemia. Okay, type 2 tyrosinemia. There is a deficient enzyme that's called as tyrosine transaminase. That's the first enzyme. Okay, tyrosine transaminase. Okay, that's the enzyme which is deficient, and there is some typical feature like corneal ulcer and ulcers on palm and soles. So this is known as oculocutaneous tyrosinemia, right? Oculocutaneous tyrosinemia. And type 3, or it is also called as neonatal tyrosinemia. Neonatal means temporary rise of tyrosine levels. It is causing no problems, right? It is like I can say a phasic or temporary rise of tyrosine levels, right? Deficient enzyme is para-hydroxyphenyl pyruvate dioxygenase. Okay, para-hydroxyphenyl pyruvate dioxygenase. Okay, right, so this enzyme is deficient, and there is a transient rise in tyrosine levels, because there are some reasons that this PHPP can enter into the other pathways and that produce some non-toxic compounds, right? So it's not a toxic condition. This has been asked once in names, the name of disease and enzyme, right? So transient, this is a transient rise of tyrosine, which will be fine. Okay, guys, there is in type 1 you can write one more line, two more lines actually. So in type 1 tyrosinemia, just add two more points. One is there is a boiled cabbage, just like hypermethylomania. Boiled cabbage odor in the patient's body or maybe in the urine. And second is drug therapy is an orphan drug, nitisinone. Okay, nitisinone is an inhibitor of PHPP dioxygenase. Sorry. Okay, everyone, nitisinone. Fine. Okay. Guys, WhatsApp is a big distraction, seriously. Okay. Switch it off. Everyone clear? So again, the order is boiled cabbage order and drug is nitisinone. So guys, that's all about tyrosinemias, three types of tyrosinemias. The second disorder of this tyrosine metabolism is L-captonuria. Okay, L-captonuria was the first of its kind, like first inborn error of metabolism diagnosed. Okay, it was the first inborn error of metabolism which was diagnosed. Okay, which was found or discovered. Okay, it is a part of Garrett's tetrad. Okay, Garrett's tetrad, which also involves lots of other disorders like pantosuria, cystinuria. This is L-captonuria. You will read in your surgery again, medicine, Garrett's tetrad. Okay, so L-captonuria. This is deficient enzyme homogentisic acid oxidase. Okay. Nitisinone is the inhibitor of PHPP dioxygenase, so it will inhibit the whole tyrosine metabolism. Okay, so PHPP will divert it into the other pathway, and that is non-toxic pathway, right? So homogentisic acid oxidase. Fine. So what happens in this case is there is increased amount of homogentisic acid, right, which appears in the urine, right, and this homogentisic acid is oxidized by the oxygen in the air, right, to the compound that's called as benzoquinone, right, which converts into benzoquinone polymerase 2, a black pigment known as alcohol. Okay, so that's why the name L-captonuria, right? So guys, this is the case where the urine turned black, okay, on the air exposure, right? So urine is not black. This is not a case of dark urine, like not a case of hemoglobinuria or hematuria, whatever, right? It is not it is a case of L-captonuria where the urine was normally fast. If you keep it for a few minutes or if you add some alkali, it will make black color, right? So urine turns black on air exposure. 1 point. Okay, also there is a high amount of homogentisic acid, and this accumulates in the soft, right, and this gives rise to the black color. So blackening or darkening of nose and sclera, okay, and the sclera. Clear? So blackening of the nose, pinna, and the sclera. Fine. Okay, so homogentisic acid accumulates in the soft tissues, right? And there's this homogentisic acid will also accumulate in the joints, right? For example, in the disc, intervertebral disc. Please write intervertebral disc, and this leads to degenerative arthritis. Okay, this will lead to the degenerative arthritis. Okay, so everyone clear? So accumulate in the joints. So I'll show you one picture, and that's for sure I have that. See this, guys. Okay, you can see this. This is the urine sample. Okay, so this is the urine sample which turns dark. It was passed normally. See the sclera between the cornea and canthus. It is black, right? It is dark. And see the joints, they have turned black. See the pinna, one of the earliest site of the darkening, right, is the ear cartilage. So these all the points were mentioned in a question in AIMS exam and NEET PG exam, right? Like the patient had these features. Even X-ray features were mentioned, like degenerative arthritis. Okay. So that question was simple because of the darkening of the urine was mentioned. So question was very simple, right? Other features were just mentioned to distract you. Okay, because when you sit with the 19 subjects in your mind, so you will not think directly of alkaptonuria. You will also look at the, let's say, X-ray feature or some other clinical points or whatever. Okay, so this is the key point: darkening of urine on exposure to air is alkaptonuria. Right? Here, guys. Okay, I will change the page now. So that is your alkaptonuria we are talking about. Okay. See, guys, one more point just I want to add here. Okay, these are, please write it. Hope it's visible. Guys, these are, this is the biopsy of the dermis, okay, skin of a patient of alkaptonuria, and it has some banana bodies. These are the homogenic acid accumulation, and this is visible as banana bodies. Okay, which has ochre color. Ochre means the color, that this is the color that's called as ochre. Okay, like the color of the clay or the pot. Okay, ochre color. So that's why this case is also known as ochronosis. Fine. This is also called as ochronosis. Right here, everyone. See this banana shaped bodies which are visible. Okay. So we can write this point, that at least you should write this point, that alkaptonuria is also called as ochronosis. Okay. Why ochronosis? That I have mentioned. Okay, why it is called as ochronosis. Okay, so ochre colored banana bodies, right? Ochre colored banana bodies, right, guys? Okay, boys are weak in colors. Okay, so remember this color also. This is also a color, ochre color. And this is why we call it ochronosis, right? Girls can easily remember. Okay. A few guys are doing hi and hello in the chat section, please. Please very much. Everyone clear? This is called as L-captonuria. Okay. So these are the disorders that you must remember. Now just write the next heading in continuation, that is products of tyrosine. Okay, products of tyrosine. So that's number one, melanin. Okay, number one is melanin. So in simple step, we take tyrosine. We take the enzyme that's called as tyrosinase, okay, which is copper dependent, and this is converted into melanin. So this occurs in the melanosomes, right, in the keratinocytes. So tyrosinase, copper dependent enzyme, deficient tyrosinase. Okay, this is called as oculocutaneous albinism. Okay guys, albinos. So albinos, they lack this enzyme that's called tyrosinase because melanin gives the color to the eyes and the skin, hairs, everything, right? Okay. So oculo plus cutaneous albinism, that is the deficiency of tyrosinase. Right guys, clear to everyone? Copper dependent enzyme. Okay, this is a copper dependent enzyme. Okay, and all the products which are marketed, especially in India, which are known by the names of Fair and Lovely, okay, or the Fair and Handsome. They contain these all the fairness products, whatever they are. They contain some chemicals which are inhibitor of this enzyme, tyrosinase. Okay, right. And so are they safe? Obviously not. Okay. See, there are no human studies as such. Human studies are in the process of completion. The people who are using it, they will tell what are the effects, side effects, right? Because in the animal studies, they are found to be very toxic. Okay, so this is the first product, that is melanin. Number two, after the melanin, we have thyroid hormones. Okay, thyroid hormones, right? So these thyroid hormones are T3, T4. Okay, so monoiodothyronine, I mean to say T3 is triiodothyronine. Okay, so it is just attachment of iodine to the tyrosine residues of the protein thyroglobulin, right? Number three is catecholamines. A very important topic that we are taking is what are the products of the tyrosine. Okay, products of tyrosine. Okay. So catecholamines, how they are formed? So again we take tyrosine. It is acted upon by tyrosine hydroxylase. I told you it is a H4 dependent. So guys, what we get is DOPA. Okay. And the next enzyme is DOPA decarboxylase. Okay, DOPA decarboxylase. Okay, which is vitamin B6 dependent. Okay, vitamin B6 dependent. This produces dopamine. right? So dopamine is one of the important catecholamine. And then dopamine hydroxylase. Okay, dopamine beta hydroxylase. Fine guys, dopamine beta hydroxylase will convert into norepinephrine. And the last is, if we add the SAM, I told you, right? SAM. So what we get is epinephrine. Okay. So what is the name of the enzyme should be? It should be methyl transferase. Okay. Right. So the only difference between norepinephrine and epinephrine is, what is the difference? One single carbon atom. That's I told you already. So norepinephrine, epinephrine, dopamine, these are the three catecholamines derived from which amino acid? Tyrosine, melanin, tyrosine. Okay, and the second is thyroid hormones, tyrosine. Okay, just we have to add iodine on the thyroid and on the tyrosine, and we get the thyroid hormones. Is it clear, guys? This is the enzyme, right, which is dependent on vitamin C and also the copper. Okay, this enzyme is dependent on vitamin C and the copper. Right? Thyroid hormones, nothing here. Thyroid hormones are derived from post-translational modification of the tyrosine by addition of iodine. Okay, tyrosine modified. 3 iodine T3, 4 iodine T4. Clear? So that's all about the products of the tyrosine. Fine. Then the next is, okay, after this we have the next one, that is phenylketonuria. Okay, so let's talk about phenylketonuria. That is PKU in the. So in the PKU, the deficient enzyme is phenyl. Okay, I'm changing the page and I'm starting with PKU. Okay, so let's take PKU. So what's the deficient enzyme? Phenylalanine hydroxylase. Right, a very important topic. Then, so if this is deficient, phenylalanine will not be converted into tyrosine, but it is converted to some other pathway, right? Other metabolic pathway, and it is converted into first ketone, that is phenyl pyruvate. And this phenyl pyruvate will then convert into phenyl acetate, just like pyruvate to acetyl CoA. Okay, phenyl acetate. This phenyl acetate leads to the musty or mousy odor in the urine, is because of which both are the question. See, mousy odor is seen in PKU. Why? Because of phenyl acetate. Okay, so odor of a dead mouse or odor of a typical odor which is musty. Musty is like the odor which is seen in the undried clothes in the season currently, like rainy season. That's called as musty.
okay, or the mouse bicondor. So why? Because of phenyl acetate. And the third ketone is known as phenyl lactate. Okay, phenyl lactate. So guys, these are the three ketones. Okay. If we talk about the clinical features, okay, then there is definitely number one, and that's called as mental retardation. So the baby is not achieving the milestones. Okay, so the baby achieves milestones every like say month, in the initial stages of life, as you all know, like holding the thumb or gripping the pencil or whatever. So like these, so mental retardation, low IQ. Why mental retardation? Why? Because guys, the large neutral amino acids. So amino acids which are neutral, I told you, except 5 all are neutral, and large means bigger in size. So especially phenylalanine, tyrosine and tryptophan. Okay, so this is called as large neutral amino acid. There are others also, okay, like leucine and isoleucine, etc. So these three amino acids they are important here, transported to the brain via a common transporter, okay, that is large neutral amino acid transporter 1. That's not important. Okay, what is important is they have a common transporter at the blood-brain barrier. Okay, phenylalanine has maximum affinity for this. Okay, phenylalanine has maximum affinity, right, for LAT1, right. Also in the PKU there is very high amount of phenylalanine, okay, which saturate LAT1 transporter, large neutral amino acid transporter, LAT. Okay, so which saturates LAT1. So because of this, inside the brain currently, in this case, there will be decreased in tyrosine. So they will be decreased in very important neurotransmitter dopamine. Fine. There is increased in tryptophan, that I'll take later. So there is a decrease in the level of serotonin. Okay. Decreased tyrosine, decreased dopamine, and decreased tryptophan, decreased serotonin. Right. So because of these two important neurotransmitters, this will lead to low IQ due to the deficient neurotransmitters. Okay guys, deficient neurotransmitters. Everyone clear? So why there is low IQ levels? Okay, so mentally retarded. Okay, and the second thing in the clinical feature, that they are unusually fair. Okay, that is reaching up to the mark of albinism, but they are not exactly albinism, they are very fair. Okay, so I can call this hypopigmentation. So mentally retarded, blue-eyed, fair girls, especially because this is have more common incidence in girls. So females. So previously it was said like this, mentally retarded, blue-eyed, blondes. Okay, hypopigmentation and mental retardation. Okay, so hypopigmentation why? So this is nothing but unusually fair child, especially females, and guys, because there is high amount of phenylalanine, okay, and this inhibits via allosteric mechanisms the tyrosinase enzyme. Okay. Here, am I going fast really? Or are you guys sick? Okay, so hypopigmentation, unusually fair child. Why? Because high amount of phenylalanine inhibits the tyrosinase enzyme. Okay, so because of there is decreased in the melanin. Clear to all? That's why the hypopigmentation. So guys, these are the two important clinical feature here. One, I told you the baby's diaper or the baby's urine smells of mouse. Okay, right. So rest of the things I told you, there is mousy odor. Okay. Then there are some tests, lab test. Okay, number one, phenylalanine levels more than 20 mg per dl, which is very high. Normal is less than 1 mg, 2 mg per dl. Okay, that's very, very high. Okay. Then how will you blood should be collected? Okay, heel prick, okay, or the dried blood spots. Guys, what is this term? If you don't know, then I'll explain. Heel prick, simple, you will see during your peach posting, you will see heel prick samples can be taken from the kid. Okay, so prick the heel and you will get the fresh capillary blood. Okay, for testing, easy, right? And the sample can be taken on a tissue paper kind of thing. That not tissue paper exactly. What do you call that? The paper that absorbs. Simply like a tissue paper kind of thing. There are some papers available. Okay, so you can take the blood on that paper. Okay, absorbing paper, and all the blood, right? All the blood on that paper will be now converted into a spot. Okay, and it will dry. So this you can transport or you can send this to the lab, which may be far away. Right? That is the case like in some countries where the labs, specialized labs are available very far, only in the big cities. Okay, right, including USA. So dried blood spots. Okay, there was a question in exam that dried blood spots can be used for the diagnosis of. So guys, you can mark any answer which is a inborn error of metabolism. Okay, right. So for any inborn error of metabolism, and these are one more thing, should be collected after 24 to 72 hours of birth. Here you want to know the phenylalanine levels. Okay, right. Heel is a like easy source of the high amount of. There's a good capillary circulation in the heel, right? So basically, right. Okay, fine. Okay, the technical team, you are listening, and some students are unable to log in. Please take care of that. Okay. So guys, should be collected after 24 to 72 hours. Why not as soon as birth? Because the protein metabolism will start after consuming the milk. Okay, the breastfeed. So it will take some time to know whether the metabolism is normal or not. Okay, so blood sample should not be collected as soon as after the birth, because that may have the normal levels of phenylalanine, because the baby was totally dependent on mother before that. Now the baby will start their own metabolism, right? So should be collected after. Very important point. Very important point. Okay, number 3 point. Okay, in the same flow chart, and these are all analyzed by the tandem mass, this is the name of technique, spectrometry. Okay, tandem mass spectrometry. Okay guys, fine. So this TMS is MCQ, right? Okay, that is a question that tandem mass spectrometry is used in screening and diagnosis of all type of inborn error of metabolism, right? Okay, so used in the screening and diagnosis of all type of inborn errors of metabolism. That's called as tandem mass spectrometry. Okay, so this is a technique nowadays in India. It is at least available in almost all the big state-run hospitals, right? The medical colleges, the biggest colleges, they have this. Sorry. Very important question. It is also used for, so we cannot go into much details of TMS, right? So TMS I am covering here for discovery of new biomarker. What is the technique used? TMS. Okay, and there it is also used for dope test or drug monitoring, can be used for that also. Okay, so the athlete's dope test is also done with the help of mass spectrometry with some attached other technique, but this is the main technique. Okay, so this is called as tandem mass spectrometry. Okay, and this is the TMS is based on mass divided by charge ratio of molecule. It can be any molecule, not just protein or amino acid or lipid, anything. Okay, so ratio of the molecule, TMS, if asked in future. Okay, so till now the question which is highly repeated is that which technique will you use for the screening of, or let's say PKU, tyrosinemia, alkaptonuria, maple syrup urine disease, okay, Hartmann's disease, etc., etc. Okay, so that will be the answer. TMS, D6PD deficiency, you can do that. Okay, so that's your inborn errors of metabolism. Right guys? Okay, so tandem mass spectrometry. Some old test names only, because sometimes examiners just write them in the MCQ, and that means in the recent names. Guthrie's test, which is not used nowadays, very time consuming test. Just remember the name. Guthrie's test and FeCl3 test. Okay, so FeCl3 plus urine, and this gives the green color. Okay, so urine which has phenyl ketones. Okay guys, most confirmatory is always TMS, right? Testing the levels of phenylalanine by the tandem mass spectroscopy. Most confirmatory can be definitely the genetic tests, which are very difficult, very costly, not easily available, right? Rarely few labs conduct them around the world. I'm getting a point. Like samples, if you want to test it from India, you have to send the sample to some other country like maybe Singapore. Okay, so that's your older test. Genetic test still not very commonly available in India, right? Directly not available. So guys, that's your PKU, very important topic. Treatment part. Okay, treatment part is phenylalanine restricted diet. See the word restricted, not zero. Student mark the answer like true regarding PKU. Phenylalanine zero diet. No, not zero. It's a essential amino acid. You cannot make it zero. So it is restriction, like instead of 100 you are giving suppose 40. Okay, then phenylalanine restricted diet should be started within 7 days of birth. Okay, to prevent low IQ or brain damage. Okay, that I explained. So it should be started ASAP. Second thing, should, as per the current recommendation, should be continued for lifelong. Okay, very important point. Should be continued for lifelong, and special care if the PKU patient is planning pregnancy. Okay, so that means zero. Then it has to give almost for some time when the brain growth is active. Zero phenylalanine levels may be given. Okay, so guys, that is the points on treatment part. Okay, then BH4 analog, okay, which is approved by FDA, known as sapropterin, okay, with the only one trade name, that is KUVAN, Kuvan. KUVAN, this is the trade name, which is the commonly used name, right? It is given. Why sapropterin? Why BH4? It is the activator of the main enzyme. And the latest therapy is, okay, which is recently approved by FDA, is Palynziq. Okay, this is the name given, Palynziq. So what is Palynziq? It is recombinant DNA technology based enzyme therapy. Enzyme therapy. What is the name of enzyme? Phenylalanine ammonia lyase. Guys, these two names you should remember. Palynziq What is Palynziq? It's a new therapy which is based on enzyme. What is the name of enzyme? Phenylalanine ammonia lyase. What it does? It will convert all the high amount of phenylalanine to the ammonia. Okay guys, so this will convert all the excess phenylalanine. So this is the latest recently launched therapy, Palynziq, which will be crystallography is used where? What you are asking? Crystallography is not used here. Mass spectrometry we are talking about. Okay. So guys, that's all. So every possible detail that you should know about PKU, because this is one of the favorite topic of examiners I have given you, right? Almost everything. If you find anything extra, please add it here only. What is the best way to complete your notes? If you find anything extra during your grand test session or class test session, or maybe any reading you are doing from other book. So if you find an important point that you think is asked as an MCQ already, so you should add it in the notes. So that's why while making notes, you should keep some space between the two topics. Okay. So like when I start PKU and if I start now tryptophan, you should keep some space. If you find any important update, you should add it at the same place, because you cannot make a separate notebook of updates or important questions that I found. No, just same booklet. Okay, otherwise it will be very, very difficult. Treatment side is nothing. This is Priyadarshini. This is Palynziq, sapropterin, that is BH4 analog. Okay, then we should start the treatment with the restriction of phenylalanine. We should be started within 7 days of life. Okay, and should be continued for lifelong. That's it. Okay. And also, now one more point that you should know is, okay, you can also supplement tyrosine, because now it's which is now a conditionally essential amino acid. Okay, so that will be the conditionally essential amino acid. Very important term, you must be knowing already. Conditionally essential. Now this becomes essential in case of PKU, otherwise it is not. Okay, fine, right. So that's all about PKU. Now we'll start with a small topic, that is tryptophan. So one more aromatic amino acid. First of all, tryptophan products and metabolism. So metabolism is very, very big. Just few points that you should know. We'll start with transaminase. With multiple steps, we generate something known as skin uranin. Okay, skin uranin. Fine. And this skin uranin is acted upon by an enzyme that's called a skin uraninase. Okay, which is vitamin B6 dependent. Okay, vitamin B6 dependent enzyme, and again by lots of number of steps. Okay, number of steps. What we get is the NAD. which is not the free niacin. Okay, not free, but it is niacin. So what is this? We will call it definitely niacin or nicotinic acid, but it is not free. It is the active form of niacin, that is with the nicotinamide adenine dinucleotide. Okay, so what we get is NAD. Fine. So guys, here are some points which can be important, which are important. Okay. First of all, there is a condition which is called as HART-NUP, Hartnupp's disease. Right? Hartnupp's disease. Hartnupp's disease is a type of amino aciduria. So this is a this is defective neutral amino acid transporter SLC6A19. Okay, SLC6A19. Hello. Yes. Thank you. Okay, SLC6A19. What's the name of transporter? Done. Sapropterin is BH4, nothing else. Somebody is asking sapropterin is activator. So it's same as BH4, no? BH4 analog. Done, everyone? Okay. Kinurenase makes NAD or niacin. So Hartnupp's disease, defective neutral amino acid transporter SLC6A19 is a solute carrier. SLC, name of the solute carrier. Okay. So it's a case of amino acid urea, right? It's a case of amino acid urea, right? So all neutral amino acid except glycine and proline. Okay, glycine and proline, which has separate transporter. Okay, so all the neutral amino acid except some like the glycine or the proline, they will appear. Okay, so they will appear in urine, right? Especially with unknown reason, high tryptophan. That's not known why, right? Why tryptophan is most importantly removed, and why not there is a deficiency of other amino acids, especially the tryptophan deficiency will be seen here. Okay, and plus there is decreased absorption of these amino acids and especially tryptophan, because I told you this is okay, I have not told you, this is the transporter which is present in kidney plus intestine both. Okay, it is present in kidney and intestine, so both reabsorption and absorption are affected. Okay, reabsorption and absorption both are affected here. Remember this, it is present both in the kidney and intestine. So decrease in absorption in the intestine Okay, so this will lead to especially the tryptophan deficiency. Okay, and because of this leads to pellagra-like dermatosis. So guys, you all know the pellagra-like dermatosis. Okay, pellagra-like dermatosis. Why? See, decrease in tryptophan, decrease in niacin. So that's why. Okay, everyone clear? So why this case is presenting just like pellagra? Okay, so pellagra-like dermatosis. So decrease tryptophan, decrease niacin. That's the reason. Okay, so when the niacin is deficient, what is the condition known as pellagra? And in the pellagra, what is the special of dermatitis? That you know, in the pellagra, the dermatitis looks like the castle's necklace. Okay, like this. Okay, so it looks like the sunburn around the neck, which looks like a necklace. Castle is the name of Dr. Castle who found it. Castle, C-A-S-A-L, necklace. It recently asked image-based question, which is seen in pellagra and which may be seen in Hartnupp's disease, which is a rare disease, right? Pellagra also nowadays become rare because of the various programs which supplements the nutrients, okay, around the world. Okay, fine. So food fortification, that is adding nutrients to the available food and decrease the incidence of lots of disorders around the world. Pellagra is also one of them. Still in Asia, you can find these Africa and Asia cases can be seen. Okay, so pellagra-like dermatosis is seen. This term is highly important. Please remember this. This is the second name of Hartnupp's. You can remember like this. Okay, and this patient also presents with some mental neurological problems like cerebellar ataxia. That may not be important because your whole question will be based on this point: pellagra-like dermatosis. Okay, and SLC 6A19, presence of all the amino acids in the urine except, I told you what. Otherwise, you will read in your probably pathology part, on the medicine part, something about Fanconi's syndrome. Okay, as in the Fanconi syndrome, there are a lot of things appearing in the urine, including all the amino acids, right? Phosphate urea, bicarbonate urea, etc., etc. But in this case also, most of the amino acids are there in the urine, but not the acidic, not the basic, even not all the neutral. Clear? So remember, this may resemble like Fanconi's anemia, but it is not, because there is nothing like phosphate or bicarbonates in the urine. Okay, and special point is pellagra-like dermatosis. That's it. Okay, and guys, there is a in the deficiency of vitamin B6, there is xanthurenic acid urea. Now see this point first of all here. So when the kin urinin is converted into niacin, it needs vitamin B6. Yeah. So in the deficiency of vitamin B6, okay, so if there is a vitamin B6 deficiency, this converts into a compound that's called as xanthurenic acid. In continuation, xanthurenic acid. Okay, everyone. So guys, this is a test of which was previously used, but still important for MCQs. So xanthurenic acid urea is seen in deficiency of vitamin B6, tryptophan metabolism disorder. Okay, and tryptophan load test. Okay, so if you give a patient oral tryptophan and urine sample, after the 2 hours you collected the urine sample. So urine sample has xanthurenic acid. So this is the old test nowadays, diagnostic of pyridoxine deficiency. Right, nowadays we do some other tests like transaminase test, erythrocyte transaminase test. Okay, so diagnostic of vitamin B6 deficiency. What do you call this? Tryptophan load, or you can say xanthurenic acid test, or you can say xanthurenic acid urea occurs in which vitamin deficiency? Vitamin B6. Okay, vitamin B6. So, what are the products of? Yeah, we will take the lunch break now after just last point. Okay, what are the products of? Okay, products of tryptophan. So, what are the products of tryptophan? You can remember them. So, guys, basically serotonin and melatonin. These are two. One is serotonin, that you all know. What is that? Melatonin, you all know. Sleep hormone, pineal gland. Okay. And so how it is taking places, like you start with tryptophan in certain tissues. The name of enzyme is known as tryptophan hydroxylase. Okay, tryptophan hydroxylase. Right. This is again BH4 dependent. And I told you. Okay, this is converted to 5-hydroxy tryptophan. Okay, 5-hydroxy tryptophan. Fine. Then 5-HT decarboxylase. Okay, guys, all the decarboxylases are vitamin B6 dependent. Okay, every decarboxylase is B6 dependent. All the transaminase are B6 dependent. Okay, can you answer me the question, which enzyme of carbohydrate metabolism is B6 dependent? So that's your homework in the lunch break. Which enzyme of carbohydrate metabolism is dependent on B6? Only one, right? Which is specifically involved in carbohydrate metabolism. Okay, directly in carb metabolism. Okay, so 5-HT decarboxylase, this produces 5-hydroxy tryptamine. Okay, 5-hydroxy tryptamine, which is also known as serotonin. So once this question was asked, like, what is the chemical name of serotonin? Which of the following is serotonin? Tryptophan or tryptamine? Tryptamine, not this one. Okay, and in the end, we will add SAM, donor, and we will also add acetyl coenzyme A. Okay, right? So what we get is known as melatonin. Okay, which can be written like this: acetyl tryptamine. Okay, right, methoxyacetyl tryptamine. So we have added two components, acetyl CoA and SAM. Right, acetyl CoA and the SAM. So guys, what we get? Melatonin. 5 and serotonin. Question was like this. Which of the following amino acid is associated with the maintenance of sleep-wake cycle? So what is your answer? Tryptophan. Amino acid associated with sleep-wake cycle, that is melatonin. I'm going to say tryptophan. Fine, tryptophan. So guys, you can see one point that BH4 is important everywhere in tyrosine metabolism, phenylalanine metabolism, tryptophan metabolism. So as there is a term which is called as malignant PKU, right? Which was previously very disease with high mortality and no diagnosis at all. Like what is happening? We are restricting the diet, still the baby is not responding. So malignant PKU is actually can be due to defective BH4 synthesis. Okay, defective BH4 synthesis, and or it can be due to defective dihydropterin reductase. Okay, defective dihydropterin reductase. Fine, which converts BH2 to the BH4. I told you now, BH2 is made from BH4 and PKU. I told you. Okay, so malignant PKU, what will happen? So the phenylalanine why? Because and tryptophan also no serotonin. Are you getting a point? Okay, so if there is no BH4 available, right, you diagnose the case as phenylketonuria. Fine, you thought there are high levels of amino acid phenylalanine, but then you started with only one therapy, and that is phenylalanine restriction. But you have not provided the patient BH4 analogs, which is nowadays provided almost to every case, or at least most of the cases. Why? Because that BH4 is also required for making dopamine and the serotonin from these two amino acids. So there will be no improvement, or there will also be the same case, deterioration of the IQ levels will be there, still there, even if you are not giving phenylalanine to the patient. So that is called as malignant PKU. Am I clear? BH4 deficiency. Right, this question was asked previously, not asked nowadays, but otherwise PKU is important. Right, maybe asked again, because nowadays such cases are less because of better understanding of metabolism. Okay guys, see you after the break. Take a lunch break. Lunch break will take for 40 minutes. Fine now, 30 or 40? Okay, I'm giving you 40 minutes. Right? Go and have the lunch. Welcome back. It's black. No, it's not black. It's nearly black. Okay, so let's continue. So our next topic is what we discussed is also the tryptophan is covered, right? Okay, then next is the branched. Okay, I'm checking on the next page. Branch chain. Okay, amino acid. Okay, branch and amino acid. So there are three, that is valine, isoleucine and leucine. All are also hydrophobic. I'll take one example of leucine, right, which is purely ketogenic. Okay, so if we start with leucine, okay, so the first enzyme is always the transaminase. So we have discussed like tryptophan transaminase and it was like tyrosine transaminase. But guys, have you noticed we have not used something known as phenylalanine transaminase, because phenylalanine is not metabolized much by this pathway of transamination. It is mostly by the hydroxylation to tyrosine. Okay, so leucine transaminase. Okay, then this is converted into some alpha keto acid. So if this is an amino acid, this will convert into some alpha keto acid. Okay, alpha keto isocaproic acid is the name. That may not be much important, like, okay, but what we get is alpha keto acid. Okay, now the next enzyme is BCAA, branch chain amino acid. BCAA. Okay, alpha keto acid. Okay, alpha keto acid, right? Dehydrogenase, right? So branch chain amino acid, alpha keto acid, dehydrogenase. Fine, dehydrogenase. Is that clear? So guys, this is converted into, so it can be any branch in amino acids, alpha keto acid. I am just taking leucine, so the name is common. Okay, name is common, right? So in this case of leucine, what we get is known as isovalyl coenzyme A. Okay, so what we get is isovalyl coenzyme A, IVC in the short, right? So isovalyl coenzyme A dehydrogenase. Fine, guys, this is an enzyme which is FAD dependent. Okay, can you name an enzyme which is FAD dependent that you have already done in your Krebs cycle? Who will answer? So this is FAD dependent, or the riboflavin dependent enzyme. So isovalyl coenzyme A is now converted to the isovalylic acid. Okay, okay, I'm just making it a little more short. Isovalyl coenzyme A dehydrogenase, and now what we get is known as the HMG coenzyme A. Okay, so first we'll cover the pathway leucine. I am taking all the branch in amino acid, but I am taking one example of leucine instead of taking all three. Okay, because there are some extra questions on leucine, but there is no separate question directly on others valine and isoleucine. Okay, that's why. So don't waste your time, just read this what I'm telling you first. So BCAA, so first is transaminase, then makes the alpha keto acid, then alpha keto acid dehydrogenase, then isovalyl coenzyme is made. The dehydrogenase of that will act. Okay, it will also be a decarboxylation. Okay, and what we get is known as HMG coenzyme A. Okay, leave the CO2. Now guys, HMG coenzyme A is also part of cholesterol and ketogenesis, right? Fine. So this HMG CoA lyase. Okay, HMG coenzyme A lyase, and this obviously makes the acetoacetate, which is a ketone body, plus the acetyl coenzyme A. Okay, so plus the acetyl coenzyme A. Clear to all? So acetoacetate plus acetyl coenzyme A. Fine. So guys, this is purely ketogenic amino acid. as you know. Okay, purely ketogenic amino acid. Fine. And now there are some important points that you should know, which has been already asked as MCQs. Okay, so number 1, starting from the last, let's say HMG-CoA is the intermediate in, so as you know, cholesterol synthesis, as you know, ketone synthesis. Okay, and as we have seen, leucine catabolism. Okay, leucine catabolism. Fine. Then it was recently asked that you can easily answer that HMG-CoA lyase, okay, is common to, you can answer common to which two pathways? You can easily answer leucine catabolism and HMG-CoA lyase in the cholesterol or ketone. Answer is cholesterol. Okay, answer is cholesterol. Oh, I'm sorry, ketone synthesis. HMG-CoA lyase. Let's make the acid same now. Right, so it was a very simple question asked in PGH and NEET exam. This question has been asked in NEET PG. HMG-CoA is not an intermediate in. So these three are correct. So fourth will be the answer, right? Not an intermediate in, right? So like that. So HMG-CoA lyase is common to. Synthase is only in cholesterol synthesis. Okay. Question number 3. So yeah, I asked you the in the morning. I asked you before the lunch. I asked you which enzyme is basis dependent in carbohydrate metabolism, glycogen phosphorylase. Then I asked you what is the FAD dependent enzyme of Krebs cycle, succinate dehydrogenase. Okay, right. So now the next is question number 3 is maple syrup. Maple syrup urine disease. So urine has a smell of the caramel or the maple syrup, which is a plant-derived syrup, as you know, maple syrup. So urine has a typical order of that. Okay, typical order of this particular maple syrup, which is a sweet syrup, just like caramel. Okay, so it is also urine. First of all, deficiency of which enzyme? Branch chain amino acid alpha keto dehydrogenase. This is the name of enzyme deficient. Okay, so because of this enzyme deficient, what happens is, okay, this patient will accumulate the BCAA and its keto acids, which are typically neurotoxic. Okay, neurotoxic, right? So this patient presents with a typical feature, that is, episodes of flaccidity and hypertonicity, right? So this patient has a typical presentation, presents with the episodes of flaccidity and hypertonicity, right? Means muscle tension will suddenly be dropping and then again it is increasing. I mean to say, it's just like that will be rising. Okay, fine. Okay. Fine, so this is accumulation of BCAA will be there. Okay, and its keto acids, which are having a typical feature, right, that they will have boxing or the paddling of the cycle, that kind of stance. Okay, right. So just that may not be important, but what is important is the name of enzyme, very important. Okay, then there is biomarker. What is the biomarker of this? That is a isomer, that is allo-isoleucine. Okay, allo-isoleucine. Guys, allo-isoleucine is nothing but it's an isomer of. Okay, it is the isomer of what? Isomer of the isoleucine. So guys, this is a isomer and this is typically a biomarker of MSUD. Fine, right? So detected by tandem mass spectrometry. Okay, so that's the biomarker. Okay, then so guys, one more point that you already know is branch chain amino acid alpha keto dehydrogenase, right, is catalyzing oxidative decarboxylation, right? Okay, so just like decarboxylation, and is dependent on the 5 coenzymes, right? 5 coenzymes of PDH. Anyone? What are the 5 vitamins required by the PDH complex? 5 coenzymes. Okay, so that is vitamin D1 or the thiamine, right? So I can write like thiamine pyrophosphate, B2 or the riboflavin FAD, okay, B3 or the niacin, that is NAD, fine, pantothenate or coenzyme A, that is active form of vitamin B5, okay, and definitely not a vitamin but lipoic acid. So guys, these are the 5 coenzymes which are same as, okay, so same as PDH and alpha ketoglutarate dehydrogenase complex, okay, or TCA. So PDH complex and alpha ketoglutarate dehydrogenase complex. Now there are the questions like this. See, the question is like this: which of the following enzymes catalyze oxidative decarboxylation? Okay, removal of the carbon dioxide with the generation of NADH. Right? Okay, generation of NADH from the removal of hydrogen, that is oxidation. Okay, so answer is 3: PDH, alpha ketoglutarate dehydrogenase complex, and this one. So which enzymes catalyze the oxidative decarboxylation? So your answer is 3. Okay. Then what are the vitamins of Krebs cycle? Same. What are the vitamins required for branch chain amino acid metabolism? Same. What are the vitamins required for pyruvate to acetyl CoA conversion? Same answers, right? Same 5 answers. Everyone, same 5 answers. Very good. Okay. Raji Gupta, right. Okay. So these are the same. They will all be same. Clear to all? Fine guys. These questions are very common. 3 enzymes and these 5 coenzymes. The name should be there in your mind always. Okay. Very, very repeated question, and that's why. Okay. MSUD can be treated with thiamine. Acha, if I ask you like what is the most important vitamin out of all? Thiamine. Okay, because it is catalyzing decarboxylation oxidative decarboxylation. Now see the difference. Oxidative decarboxylation means there will be generation of NADH that I didn't mention here. Okay, there will be definitely generation of NADH. But when I say decarboxylation, it is B6 dependent, decarboxylase. Okay, like I told you, 5-HT decarboxylase makes the melatonin or serotonin. Okay, so like that. Clear to all? So MSUD can be treated with the thiamine. Okay. E1 alpha subunit of branch chain amino acid alpha keto dehydrogenase complex is TPP dependent. Just like E1 of the PDH is TPP dependent. There are three enzymes in PDH now, E1, E2, E3. So E1 is dependent on enzyme number 1, like pyruvate dehydrogenase there, is dependent on thiamine. Similarly, it is E1 alpha subunit, it has two alpha and beta subunits. Alpha is dependent only on B1. This is also a MCQ that you will find during your practice. Clear? So which vitamin can be used to treat MSUD? All, but most importantly thiamine. All the 5, but most importantly thiamine. Clear guys? Everyone? Okay, so now the next point is so this is all about the MSUD. It is difficult to treat. Why? Because all 3 are essential amino acids. So it's very difficult to treat because all 3 are essential amino acids. Okay. There is a condition that's called as deficient IBC dehydrogenase in the above case. Okay, and this is called as isovaleric acidemia. The only thing that you should know is a case of metabolic acidosis. It is metabolic acidosis, and as this metabolic acidosis presents with a typical feature that has been a part of question, and that is sweaty feet odor. Okay, in the body of the patient or urine. Okay, sweaty feet. Okay, sweaty feet odor. Clear to all? So isovaleric acidemia. Just at least remember the disease name, enzyme, leucine, and order. Right, there's some of the important products. So, like as amino acid, arginine, and its product is nitric oxide. Okay, nitric oxide. Anosynthase is the name of enzyme. Okay, so what's the name of enzyme? Nitric oxide synthase. Number 2, glutamate. Okay, and this is converted into GABA, glutamate, okay, gamma amino butyric acid. Name of enzyme, glutamate decarboxylase. Okay, so as you all know, nitric oxide is a vasodilator, fine, potent vasodilator, and GABA is a neuroinhibitor, right? Neuroinhibitor. So this is vitamin B6 dependent enzyme. Okay, and that's why the deficiency of B6 can lead to neonatal seizures. Why? Because guys, the neuroinhibitory substance is deficient. MCQ was in the recent JIPMER exam. This was a question. Okay, around 1 year back. Fine. So, deficiency of which vitamin can lead to neonatal seizures? That is vitamin B6. Why? Same. This is the answer. D-carboxylase is dependent on that. GABA is a neuroinhibitory substance. Okay. Number 3. Histidine is converted to the product that is histamine, which is mediator of inflammation. Okay, bronchoconstriction. So histamine, fine, and this enzyme is called as histidine D-carboxylase. Okay, histidine D-carboxylase. Clear it all? So that's the histidine to histamine. Like this. Okay. Histidine is also associated with histidine act as buffer. Right. So which is important part of hemoglobin buffer. So guys, as you all know, hemoglobin also acts as buffer, right? And the only amino acid which has a pKa dissociation constant, the pKa value which is nearest to the blood pH, and that is histidine. Okay, it is around 7. Right, the pKa, the dissociation constant of the histidine where it is ionized, okay, it is around the pH of 7. That is near to the blood pH. So at the blood pH, hemoglobin can act as a buffer to maintain the acid-base balance, and histidine is very important in that. Okay, histidine is important in Figlu test, or it is also called as the histidine load test. Okay, histidine load test. Okay, for the diagnosis of folic acid deficiency, not used nowadays, right? But you should know this. Find histidine load test for diagnosis of folic acid deficiency. How? See guys, the load means orally. So first of all, patient is given oral histidine. Okay. Then this oral histidine metabolism. Okay. Name of enzyme is histidase, not important. Urocanic acid, not important. Okay. This urocanic acid is converted into formyl amino, okay, glutamate. Okay, that is called as Figlu. Okay, that's called as the Figlu. So this is the name of the test, Figlu test. Okay, Figlu, Figlu, formyl amino glutamate. And to convert this Figlu to the glutamate, so glutamic acid is made, and for this we need tetrahydrofolate, which is converted to the formyl tetrahydrofolate. Am I clear, everyone? Okay, so this is your this is how the this Figlu test is conducted. So what is Figlu test? Histidine load test. So guys, this is a question which has been repeated number of times, but nowadays these tests are not done, but still questions are being asked, right? Okay, nowadays you can directly measure the folic acid levels, isn't it? You can directly measure the folic acid levels. Okay, so moving on to our next topic. Okay, so guys, first of all, we have a disorder or amino acid which is called as glycine. Okay, glycine. So guys, glycine is metabolized by the glycine cleavage system. So we take glycine plus NAD plus hydrofolate. Okay, and this enzyme is called as glycine cleavage system. Cleave it, take it. Okay, GCS, and this produces CO2 plus NH3 plus the carbon. One of the carbon is given as methylene hydrofolate. Okay, right. So guys, glycine has a role that in this way. Okay, so glycine act as one carbon donor. You can see here one carbon donors. Okay, understood? So this is the one point. Now there is first of all, what are the products of glycine directly? Products of glycine, guys, we have number one, heme. So heme I'll take after this amino acid. So heme, you will see how it is made with the help of this. Number two, purine ring. So adenine and guanine ring. Okay. Number three, glycine is required for synthesis of glutathione. Okay, glutathione. Glutathione is made of 3 amino acid. Okay, it is a tripeptide, right? Which is made of gamma glutamyl cysteine glycine. See guys, glutathione now, if you write like this, अगर आप ऐसा लिख लो उसको GSH, so you will never forget the 3 amino acids of glutathione. You know why? This is the chemical symbol of glutathione. It is written like GSH. Fine. So you can easily remember GSH like this. So G, first of all, SH. SH groups are only present in cysteine. Okay, cysteine. So what are the 3 amino acids? Very important question in the GSH. Cysteine. G for the glycine and G for glutamic acid. Only 2 amino acids we have from G. Any other you know? Glutamate and glycine are the 2 important amino acids that we have. Okay, you got the point, everyone? So glycine plus glutamic acid, G, and SH for cysteine. Okay, cysteine has SH groups. Number 4, creatine. Okay, creatine. Achha guys, glutathione, as you all know, what is the function of glutathione? Glutathione is, okay, as you all know, and if you don't know, we'll discuss that again. Is your antioxidants done in the class? Anyone? Antioxidants done or not? GSH is the antioxidant and it also helps in the transport of amino acid, okay, across the cell membrane. So it's not just only antioxidant. No, not done. Okay, antioxidants, fine. So creatine. Now next is creatine. Remember two functions. GSH is antioxidant and GSH is also important in transfer of amino acid across the cell membrane. Okay, that you will read in your renal physiology. Okay, but I'm just writing here transport of amino acid. Okay, then the next is creatine. Okay, so guys, creatine. So do you know guys, creatine is something which gives us energy during muscle contraction. Okay, the bodybuilders, athletes, they take creatine supplements, to increase their muscle power. How? You know how? It is very simple, right? Okay. Creatine, first of all, it's again a tripeptide made of. We can remember it like this: GAME. Okay, so G for glycine to hona hi hai. Now we are talking about glycine. So glycine, A for arginine, ME for methionine, which is actually SAM. So SAM is also required for creatine synthesis. Okay, so guys, why I have written GAME? Remember this, you can draw a athlete or a runner or a bodybuilder, right? Just to remember GAME. Okay, GAME means exercise. Okay, so creatine is required for playing games, exercise. So glycine, arginine, methionine, and methionine is SAM. Okay, so guys, creatine, it's not creatinine. Okay, creatinine is based of creatine. So creatine is the phosphocreatine. Phosphocreatine is the storage of high energy phosphates. Okay, and this is the fastest source of ATP generation during exercise. Right? How? We take simply ATP, okay, and we convert this into ATP. Name of enzyme is CK or creatine kinase. Okay. And this creatine kinase will just take, from the creatine phosphate, which is a phosphate donor. Okay, and kinase transfers the phosphate. You learned here. So, what is the fastest source of ATP generation during exercise? Creatine, which is present in our muscles normally, but that will last for only few seconds. Okay, but if you consume creatine before running, then you can definitely run for minutes, right, without getting fatigue. Okay, so that is the creatine supplement which is taken by athletes during their training periods. It can help in muscle growth, okay, adaptations. Okay, so this is your creatine. So guys, I am talking about what are the products of glycine: heme, purine ring, glutathione, creatine, not the pyrimidine ring, only the purine ring. Okay, and anything else which is made of glycine. Okay, so these are the important ones, right? If you remember anything else, you can add on. Okay, now there is a condition or the disorder of the glycine metabolism, and this is called as hyperoxaluria. Okay, primary hyperoxaluria. Primary, please write primary hyperoxaluria. Okay, this is the patient presents with oxalate stones. Okay, oxalate stones. Okay, kidney stones. So patient present with that.
So in this case, actually glycine normally metabolized by the various mechanism including D-amino acid oxidase, and this is metabolized to a compound that is glyoxylate. Okay, glyoxylate. Now guys, this glyoxylate can convert into oxalate. Okay, so that's why the disease is known as hyperoxaluria, but not normally. This is with the help of oxygen. Okay, so normally what happens, this glyoxylate doesn't form oxalate much quantities, very small quantities are going through this. So this glyoxylate convert back to the glycine. Actually we just remove the ammonia, I mean to say nitrogen. So if I add nitrogen again, yes, I can add. So guys, if I add the nitrogen again, okay, so if I add the nitrogen taken from alanine, right, so I will take enzyme alanine glyoxylate transaminase, which is present in peroxisomes. Okay, present in the peroxisomes. Am I clear? So which amino acid is now donor of? So we are just transaminating glyoxylate with alanine, right? Alanine will generate pyruvate. Okay, so if I take, like, if I take alanine will convert into pyruvate and gives the NH2, right? NH2 will be given here. Fine guys, so this happens only in the deficiency of this enzyme of AGT. This is AGT. So guys, oxalate stones, okay, will only be found in the deficiency of AGT, alanine glyoxylate transaminase. Okay, alanine glyoxylate transaminase. Okay guys, okay, so this is called as primary hyperoxaluria. Okay, primary hyperoxaluria. So how the patient presents? Kidney stones, severe pain in the flank region. Okay, so that's all about primary hyperoxaluria. Fine. Now there is a special disorder, okay, which is called as rare disorder, very rare, fish order syndrome. So next topic, fish order syndrome. So guys, what is fish order syndrome? The patient smells like rotten fish. Okay, it is also known as trimethylamine-urea. Okay, so what do you call this? Trimethylamine-urea. Okay, fish order syndrome. Okay, so trimethylamine-urea is the name. That's the older name, but more commonly asked name till now. But recently they have asked this name 2 to 3 times in the recent exams. Okay, so guys, what is this disorder is? So let's understand this. What happens? So normally we consume proteins. So I'm taking it on the next page. It's hanging a little bit. Okay, so continue with this. So we take protein-rich diet. Okay. So guys, protein-rich diet is like fish, for example. Fine. So fish has trimethylamines.
Okay, what is fish rich in? Trimethylamines, TMA. Right, that's why fish has that typical order. Fine. Or we can take, like in our diet, it may be egg. Egg is rich in choline. Okay, eggs are rich in choline. Okay, choline is a type of amine. Right, choline is a type of amine. So even egg, even other meats, right? Chicken, then nuts. They all have, even vegetarian food has choline. So they are all rich in the choline. Fine. So what happens? So this protein in the diet. Okay. So protein is acted upon by the gut microbes. Guys, we have millions, billions of microbes in our intestine, which may be healthy, and sometimes they become unhealthy and leads to disorders. Okay, so these gut microbes are resident of our gut. Okay, and they have lots of they help to maintain our normal health. That is not currently out of focus right now, but normally these gut microbes, they act on the protein and generate trimethylamines. So fish already had trimethylamine, but they generate TMA from the choline, which is present in protein-rich diet of egg and chicken and all, right? So TMA. Now this TMA has a rotten egg order, which is not found in a normal individual. Okay, so what happens next is this TMA diffuse to the liver, okay, via portal blood. Okay, now in the liver there is an enzyme that's known as FMO3. Okay, FMO is flavin monooxygenase. Okay, flavin monooxygenase. Number 3. Fine guys. So what this enzyme does? This enzyme converts TMA to TMA oxide. So this is done by FMO3, right? And this TMA oxide is orderless. Okay. So this is not a disease. This is a condition which is a social stigma because of the typical body order, right? Okay, which cannot be relieved easily, right? Because there is no treatment as such. So treatment will include definitely. So you got the point that TMA accumulation will lead to in the deficiency of FMO3. This is called as fish order syndrome, right? So deficiency of FMO3 is called as fish order syndrome. Repeated 3-4 times in the recent exams in the last 2-3 years. Okay, very important. And one of the difficult questions they asked was based on the treatment. So now it will be easy for you because I started with that point. So avoid definitely protein-rich diet. Okay, especially no fish. And avoid choline. This was the answer in one of the MCQ asked in the PG exam. So what should be avoided? Choline. So what is choline? It's an amine which is present in high quantities in our body. But the food that we take also contains it in high quantity, like eggs and chicken. Okay, and even the fish. has little amount of it. So avoid that, right? Okay. Otherwise, there is no treatment as such. You know, one of the author of a book, he mentioned that if a person is going for a special occasion, you can imagine what can be the special occasion. So the person should take metronidazole. Why? Because that will kill the gut microbes. That will definitely, for a time being, relieve the fishy or the typical rotten fish odor in that patient's body. But that's not a long-term solution, okay? Proteins cannot be avoided. That's not again a long-term solution. Okay, nice. So that's all about fish odor syndrome. So one of the special products of amino acid I'm taking now, and that's called as Heme. Okay, so just write on the next page. So leave at least your amino acid products and metabolism is over. The one product I am taking separately is a chapter. So just write heme synthesis. Okay, heme synthesis. Heme is iron plus porphyrin. Okay, it's a tetrapyrrole ring. Iron plus porphyrins will have a structure like this. Okay, so it is a structure like this. So you can see it has 4 pyrrole rings, right? It is actually a tetrapyrrole, right? So one ring, this is a one ring, this is one ring, one and one, total 4 rings, right? And in the center there is iron, right? So you can see this is in the center, there is the iron. Clear? So that's called as a tetrapyrrole ring. Fine. So these rings will be made separately, and that's called as porphyrins. Okay, guys. Okay. So heme is iron plus porphyrin. It is a tetrapyrrole ring. Right? Tetrapyrrole ring. Fine. And the heme structure that we have, it is like this, that we have the groups which are like methyl and vinyl, right? Methyl and vinyl. Then we have like vinyl and methyl. Okay, so in one of the ring there is change in the order, so that's called as the type 3 of the heme. Okay, I'll come to that point again. Sorry. So heme synthesis iron plus porphyrins. That's a tetrapyrrole ring. So heme is present in hemoglobin, right? Hemoglobin. Okay. So that's why 85% of the heme is synthesized in bone marrow, where the hemoglobin or the electrolyte precursors are being synthesized. Okay, so hemoglobin, fine. So 85% of the body's heme will be synthesized in the bone marrow and will be present in the part of hemoglobin. Right, 15% of hemoglobin is present. Heme is present in other heme proteins. Okay, heme proteins. Okay. So that is 15% of the heme is synthesized where? In the liver. What is examples of heme proteins? CYP450. So guys, any protein which is starting with cytochrome, okay, cytochrome oxidase, cytochrome reductase, CYP450, right? CYP450 is a drug xenobiotic or the drug metabolizing enzyme. Okay, so this is very, very important in the drug metabolism. Okay, so you must have read the drugs which are enzyme inducers, okay, like rifampin, barbiturates. Okay, so they will affect the heme metabolism also. Let's see. You know how they will affect. So suppose if somebody is consuming rifampin, which is a inducer of the CYP450 or barbiturates or the alcohol, so they will induce the production of CYP450, which is made of heme. So heme synthesis will start. Okay, so any person consuming a drug which is enzyme inducer will make more heme, obviously because of the reason I explained. Okay, so we'll see its application later on. Okay, so now guys, सबसे पहले, like first of all, we'll take the basics, right? That is pathway. So let's write the pathway. Okay, and some disorders. So I am taking one by one step, or if any disorder is there. So number one, so I'm writing like this. Okay, let's write steps of heme synthesis. Steps of heme synthesis. Sorry. Number one. First of all, you should know mitochondria plus cytoplasm is involved. Okay. First and last three steps in mitochondria. So we will take first and the last three steps in mitochondria. Okay, what are the pathways which occurs in both mitochondria plus cytoplasm? You can remember the mnemonic HUG, or you can remember the mnemonic that is PUBG. Okay, PUBG for the banned app. Okay, it's banned yesterday. So PUBG. What is PUBG? P for, these are the four pathways which are using both mitochondria plus cytoplasm. If you know already, well and good. If you don't know, otherwise, it is P for pyrimidine synthesis, U for urea cycle, B for blood or heme synthesis that we are discussing, B for blood, and G for gluconeogenesis. Okay? Okay, am I clear? So, P for pyrimidines like uracil, U for urea, B for blood or heme, G for gluconeogenesis, mitochondria plus cytoplasm. So first and last three steps in mitochondria. Now the first step. So guys, we take glycine plus we take intermediate of Krebs cycle. Very important question. This is a TCA intermediate. This question has been asked number of times, and question is like this, that which of the following intermediate of Krebs cycle will be utilized in heme synthesis? Answer is succinyl coenzyme A. Right. So first step I am taking here, and the name of enzyme is ALA synthase. ALA, capitals ALA synthase. Whereas this ALA synthase is B6 dependent. Okay, vitamin B6, and this is the rate limiting step of the enzyme. Rate limiting enzyme. So guys, this produces the first product, delta amino labulinic acid. Okay, delta amino labulinic acid. Fine. Okay, that's in the short ALA. Okay. Now guys, see, this is the rate limiting step, right? ALA synthase. Okay, this allosynthase is inhibited by, so allosterically inhibition, heme itself and glucose. Okay, carbs. So these two inhibit this enzyme, heme and glucose. Feedback inhibition. What is the importance of glucose? I will tell you in the next point. But remember like this. This is the rate limiting enzyme. Okay. Deficient allosynthase. Now when I say deficient, it means mutant also. Okay, it doesn't mean deficient, doesn't mean deficient means not working or abnormally working. That is called as deficiency. It's mutant. So guys, this leads to a disorder which is the most common type of inherited sideroblastic anemia. So X-linked sideroblastic anemia. Okay, X-linked sideroblastic anemia. Now guys, to understand this point, let me show you one image. Right, first of all, see, I have started with glycine plus succinyl-CoA. Okay, we have started with glycine plus succinyl-CoA, and this was an enzyme which makes allosynthase, which makes the amino levulinic acid. Now guys, in the end, actually, okay, in the end actually we add the iron. Right, that is not shown here, but the last step of the pathway uses is added with the iron by the ferrochylidase enzyme. So actually iron was added in the last step of heme synthesis, and now first step is missing. So what will happen? See, we are adding iron in the last step of the heme synthetic pathway, and now we are right now at the first step. So if the first step enzyme is missing, obviously you cannot add iron, and this iron will accumulate in the bone marrow cells, erythroid precursors. Okay, and this will be visible as sideroblast. So what is a sideroblast? Sideroblast is nothing. Okay, it's a RBC with the nucleus, and with this, these blue dots you can see around them. So this is called as ring of iron. Sorry, ring sideroblast. Okay, so no matter how much anemias you have read in your pathology, but this is the point here that you should know that iron, why the iron is accumulated here. Understood? Okay, you can see here again number of RBCs, and this is a nucleus, and around the nucleus there are blue stained iron. Right? I'm sorry. Fine. So sideroblastic anemia, X-linked sideroblastic anemia, most common inherited sideroblastic anemia. Most common inherited. Okay, so what is this sideroblast? Okay, this is RBC with nucleus. Okay, RBC has no nucleus, so RBC with nucleus and the ring of iron around the nucleus. Okay, that's called as sideroblastic anemia, and this will be obviously just like iron deficiency anemia. So microcytic hypochromic iron deficiency, right? Just like that, but it's not iron deficiency. Microcytic hypochromic anemia, but with excess iron stores. Okay, with high iron or the normal iron stores. So it is iron not being utilized, not deficient here. Iron is not being utilized here. Am I clear, guys? This is the first step of the pathway. Second step. So this will be in sequence. You can draw it in the last. I am just taking step by step and any questions if they have. Number 2. Done, everyone? Number 2. So what is the next one? We have delta ALA, amino levulinic acid. This delta ALA is acted upon by the enzyme that is called as ALA dehydratase. Okay, so that is ALA dehydratase, and as this ALA dehydratase converts the ALA into porphobilinogen. Okay, in the porphobilinogen. Fine. So in the porphobilinogen, or in the short PBG. Whereas the question here is that this is the enzyme which is inhibited in the lead poisoning. Okay, this enzyme is inhibited by lead poisoning. So some of the books they have mentioned wrong, some notes anywhere some student finds and then asks me again, sir, is it ALA synthase or ALA dehydratase? Guys, it is always dehydratase. Just remember like this, when I write LEAD, so I am talking about D for dehydratase. Okay, so remember D for this dehydratase, just like that, lead poisoning. Okay, so in the lead poisoning you are not getting the porphobilinogen. So lead poisoning is overall very important topic. So I would suggest you, when you have time, read everything about lead poisoning from your like forensic medicine and everything, right? Okay, some other features which you should know, pathology, forensic medicine, everything. Otherwise, this is one of the very repeated MCQ. Okay, and lead poisoning overall is very repeated MCQ. Fine. Like a child was eating the wall scraping. Okay, wall scraping, the paint. So then he presented with anemia, abdominal colic, constipation, etc., etc. These are the features, and basophilic stippling. Okay, so wall scraping, and there is an elevated level of ALA. So in the lead poisoning, one of the important point is increased ALA, amino levulinic acid in blood, which is very, very commonly mentioned in MCQs. So even if you don't remember the clinical scenario, if you remember this point, this will be helping you in MCQ. Okay, guys. So porphobilinogen, right? PBG, ALA dehydratase. Right, guys. 258. Okay, so next point I will take in the next class then. Okay, because that's an important topic. So you can read acute intermittent porphyria, that I will take next. Okay, so actually the third step is like this. You take PBG and you take the enzyme that's called as PBG deaminase, which is also known as uroporphyrinogen 1 synthase. Okay, so what we get is known as hydroxy methylbilane or uroporphyrinogen type 1, which is not found in human beings, right? It is type 3, which is found in human beings. So it should be metabolized further. So we will talk about this in the next lecture. So guys, you have lecture on 7th, 8th and 9th. Okay, 7th, 8th and 9th. So we will cover that up. Okay, so see you. Right. See you next time on the 7th, and I will finish the scheme and then we'll start with nucleotide metabolism and genetics. Okay. So probably next time we'll be able to cover nucleotide metabolism plus more than half of the genetics part. Fine guys, see you. Okay, bye bye. Enjoy the weather.